for digitizing, creating these various advanced media. This workshop is going to be a little bit different and more focused on the research uses, how we're using these data, how we're using AI to extract information, how do we deal with sharing of data, attribution, and various aspects. We have a very diverse number of disciplines that are represented here, from biologists to ecologists to library scientists, computer scientists, physicists. And so it's a really broad community that we're trying to develop here and bring collaborations together. So one of the goals of this workshop, which Austin will be talking about later, is how this will all culminate into a much larger event down in the future. And so with that, I'll invite Austin to come up and talk a little bit about how this workshop will come together at the end.
Thanks, Nelson. Welcome, everybody. The challenge in bringing together so many creative, interesting people is to use this opportunity to do something special. How can we make magic? And Jill had an exercise that we participated in last time. This time it's a different exercise. And let's see what happens. So this is an exercise that is designed to be a catalyst. It is how would we describe it? It's a low stakes exercise. It is an excuse for you to have a conversation with somebody who you might not otherwise have a reason to talk to. And I just want to recognize that the motivating question, or one of the motivating questions that we've been asking around this series, has been, how can we use media technologies to position biodiversity collections for even greater relevance to science, society, broadly circumscribed to include things like business, and Earth's biota in the future? And so this is designed to be a hypothetical. That is, there's an opportunity that we're going to give you hypothetically. And the hypothetical is, if we handed you a bullhorn that was huge, what would you say? That is, how would you answer that question? So the scenario is, there's a grand international event that's happening, highlighting the value of collections for science, society, and Earth's biota in the DC area next year. The event is a combination of an expo, a conference, and maybe some other stuff. Just to highlight what we mean by an expo, you've all been to conferences. There's an exhibit hall. It's very staid. There's publishers there. This is like a step up. You've been to an expo, maybe? This is kind of there's excitement in the air. It's an opportunity to communicate something. Maybe you've been to a car show. It's the crowd that goes to car shows or not. Think about exciting interactions. So the value of collections could derive from the collections themselves, the data that they create, their science, their programming, their technological innovations, for example, related to digital media or something else. The audience for this could include,
sorry,
I didn't.
That was me. That was
Okay.
me.
Could include the collections and biodiversity research community, but could also include the general public. This is in the DC area, government agencies, funders, foundations, news agencies, students, and venture capitalists. Nelson suggested it could be somebody else, but the exercise is, again, this is low stakes, and we hope that you take this up in the spirit that it's offered. That is, this is a reason for you to talk to somebody else. Collaborate with at least one other person. We're not interested in you coming up individually with some new idea or existing idea, but instead work with somebody else to recognize the combined strengths and enthusiasms that you bring to it to answer this motivating question. You'll provide a pitch for this program element. We'll think of it as a program element because We're not saying put an exhibit in the expo or create a symposium for the conference or something else. We're just saying, do something magical, do something that is communicating an answer to the motivating question of the workshop. And if you would, please identify the audience, the motivating message or theme, delivery method, a title, as well as, if you would, please recognize companies that could sponsor or co-exhibit as an activity complementary to your program element. The delivery method could combine activities in the expo, the conference, and maybe something else. Maybe it could be related to an element of unconferencing or hackathon or makerspace or something else. A single talk or a single exhibit by a single person is really out of scope for this activity. It could be half of this group is really interested in delivering some programming on AI opportunities or 3D models or something like that. You have 5 minutes. This is at the end of tomorrow, 5 minutes to present your pitch. So think about this as you're listening today and tomorrow morning. What could you combine in these various threads to build a big impact at this hypothetical expo / conference / unconference / makerspace, whatever. But this is an opportunity for us to imagine the possible. Any questions about that?
There'll be a quiz at the end.
Your grade will be wholly based on your pitch.
Austin, are we supposed to actually stand up and make this pitch tomorrow at the end of the day?
That is the idea.
And how many, what's the ideal size for the groups?
We'll talk a little bit more about it this afternoon when we go into breaks.
Yeah, there will be a bit of scaffolding around but this is intended to grow organically. We don't want to create too much structure to it. Okay, I think I've used up all of Nelson and my time in that slide. So should I invite Jill to come up maybe?
So next, do you want to just go to the next slide?
We've taken liberties. We bolted our two time slots together, and
Perfect.
we're gonna try
And
to,
you should have a screen advancer and you'll be pointing at the computer right. Yep.
okay, I'm pointing right there. So I'm gonna start and give you a little bit of intro and cover some of the things that we'll talk about. Today, we're gonna talk about the pipeline that we're developing for analyzing data for LightningBug. In particular, multi-view text detection and virtual label reconstruction are sort of two phases of our analysis, and we'll be going through that with you. This is the button that does it. So here's the basic idea. The basic idea is you have a whole lot of images taken from different perspectives, and we want to take the information that is potentially gleanable from that stack of images and convert it into virtual labels, which will look as if you had pulled them off the pin, set them out, and taken a nice photograph of them. And of course, the reason we want to do this is that it leaves the time in the equation for stripping the labels out of the entire process, thereby speeding up and enabling a very high throughput digitization process. The idea that underpins this, no pun intended, is to capture the views from several angles in order to maximize the likelihood that at least one image will contain information about any given spot on any of the labels. And of course, the magic is to computationally combine and extract all that information to make a useful end product. And to do this, we apply machine learning, computer vision, high performance computing, and various data management techniques combined in an appropriate mixture applied to the various tasks along the line that have different requirements, different needs. This is a picture that we show of the pipeline. The bottom row is the thing that we're going to focus on for the first part. That's 6 steps that we have called out as little chunklets of the algorithm that ultimately creates the virtual labels. And I'm going to talk to you for the first few minutes about this first half, which is all about pulling the text out. I actually talked about most of the first two ones in the previous workshop. So I'm going to spend a little bit more time on that last bubble. This is what we're dealing with. So there's a wide variety of things that we end up with in our hands when we take these snapshots, these multi-view snapshots, including handwriting, actual typeset text, block printing. And so how do we do this? What do we need to do to get this process pushed forward and pull all the information out of here? Well, it starts by finding the region of interest. That is, first of all, restricting the attention of our computation for the reasons of efficiency to just the area where the label information is. So we're not spending time processing all the pixels that don't have anything in them. And we do this by selecting some little algorithm bits that detect features that are kind of text-like. It doesn't have to be a text finder. It just has to be something that is strongly weighted, finding things that are in the text space. Then we cull away things with various processes, ending up with a set of points that are indicative of the space. And then we crop around that. Approximately 1,000 by 1,000 pixels is what we target. It makes a nice little size for us. Now, what do we do with that? After the region of interest, we want to detect the text and the words on each of those images. And so the thing that we've done is we've taken the image in the upper left-hand corner, which is that cropped region I just mentioned to you, and we replicate it at 90-degree rotations. And then we feed it to a best-in-breed machine learning algorithm, which has been trained not by us, but by billion megabit corporation with lots of money and lots of data to make it so that you can find text in the middle of anything. And so we're leveraging that AI to do this problem. And then we suck all the letters out and we end up with something on the right-hand side that has given us the positions of the letters and also the grouping into words. And we use that information downstream for the next set of steps. I wanted to take this moment to say that those first three steps that I described, this is the second of the three, we have gone the next step of, after finding the algorithms, of scaling them up, that is running them in parallel, putting them on a big machine so that we can run thousands at a time. And so we've already shown that we can run 2,000 specimens, 30,000 images in a very modest amount of time on a big machine, a resource that allows us to do hundreds and hundreds of processes. So we have that fabric in place. So I'll spend the next couple of seconds on this last bit, and that is, once you've got these words, what can you do to make a useful product that gets used downstream? So our goal, of course, is to find a way to link words we find in each of the poses. And if we can find a link between those for a given word, a given meta word, M word, we have an instance that may be on any one of the poses. And if we have that information, then we know that those images, the locality is useful to connect the information in that region. And Nicola uses that downstream for those processes. So my job here is to try to find out how to get those meta words. So I start by doing streaks, which is I look for combinations of 3 letters or more that match words. And you end up with a set of candidate groupings here, street lists or whatever, and they're pretty good. But the yellow one there shows one of the problems that this limited approach has. And that is that it's possible to find matches between sets of 3, which is, we do it instead of 1 because the probability is much lower that you'll have the wrong word if you have a few characters in a row that match. Still, 3 is not enough always. And we end up having 3 separate words, Chiricahua, Mexican, and Aranthromia. So I'm So those 3 words actually get mixed up in this streak thing. So what do we do? Well, remember that we can only have 1 word in one of these meta words in each pose, at most 1 word. So we find that we have at least 3. So that means that we have a clue that says that the words that were grouped there are actually probably coming from 3 meta words. So we just pick any one of them, any grouping we want, lay them across, we seed new meta words with those 3, and then one by one, we find out each word, which column does it belong in. So I've got here a partially filled out map to show you some of the times where they automatically lay next to each other, and other times where, like in row 3, pose 3, acanth needs to be moved over. So a simple lexical analysis allows us to make that work out. And when we do that, we actually go a very long way to getting the right stuff. So even though this is a kind of a sparse and weird representation of a zillion words, we have found the important relationships that can be used by Nicola to then create the virtual labels. And so, Nicola, it's you.
Okay, so the next 3 boxes are taking this text analysis and applying it to being able to extract and find all the label information and build these virtual labels. And so the first step in here is segmentation. So image segmentation is you're grouping together portions of the region that are thought to be the same object. There are a lot of tools out there that will do segmentation. We've tried a lot of them. 2 that were pretty good. Everything I'm showing you here is a meta segment anything. There's something called mask RCNN, another meta, and these all work fairly well, but we found the meta one to be actually the fastest. So it groups together and they're colored here. So if you look up in the corner there, you see different colorings and each color is representing an object. It doesn't have any idea what the object is. It just knows that all of those things are probably from one object. And so what I do is use the text detection. And if there's regions that are text, I say those are probably labels or fragments of a label. So we collect all those pieces up. And so for each pose in the set of poses of the one specimen, we're going to get a set of label fragments for that pose. Remember, we have somewhere around 15 poses per specimen and a bunch of labels. So we get all of the pose, for each pose, we get a bunch of label fragments. And then we use these M words, these meta words, the matching, to look on each of these poses and use the matching words. So you see here marks where the green is it's on the label, the cyan color is it's not on the label, but those are matching words between these poses. And we find which words match those labels. And so those are all likely different views of the same label. And so for the collections that we've been looking at, there's up to 5 labels per pose and 15 poses. So there's a lot of combinatorics here. And so the idea is to group them together. And we usually find for each label somewhere between 3 to 12 views will contain the label information. And so the next step is to rectify, take those labels and make them, nice rectangular things, then align those rectangles and then combine them together. And in the next few slides, I'll go through each of those steps. So that's sort of the process in a nutshell of what I'm going to talk about. So from those fragments, we want to actually do a rectification. We like things to be nice and square and front viewing that we can then read. And so we cull some of the possible fragments because they're too small or, they don't align first we cull for size and then we over here, you see what are called the rectifications. So they're all nominally rectangles, not perfect, but there's lots of occlusion and cropping. So from those rectified labels, we try to align them. And then again, there's a process that eliminates some that don't align well. So you'll notice here, there's labels overlapping from other labels that were on top of this one. There's the pin. And so when it aligns, like if you take this one here, you see that it's sort of rectified the text and lined it up, but there's errors because of all the other stuff in there. But if we have enough views and enough information, when we go to combine, so here I'm looking at just taking the mean of these 8 labels here, masking out the bits that aren't labeled, and you get a pretty good reconstruction of the content of the label. And so right now, we're evaluating 3 different approaches to do the alignment. They're all nonlinear alignment tools. So right now we're comparing them. I don't have an answer yet. I'll talk a little bit about that in a minute, but we're looking at 3 different techniques. They all give reasonable results. Depending on the label and the content, there's no clear winner yet, but stay tuned. Next year, I'll have a winner. Okay, so once we have the labels aligned, if they align really well, it's pretty easy. We can use trivial tools like taking the average or the median. I looked at max and min, and they work pretty well if there's not too many labels, but the goal is to use as many labels as you can. So if you have like 10 labels, The pin tends to be the minimum, and so you end up just seeing lots of views of the pin. So we're pretty much decided that the average or the median are the winner. It's really now focusing on this alignment step. So once we have them all aligned, we can do these very straightforward operations and get pretty excellent, that's these views here. This is this label here, and up there is this one. We get pretty good reconstructions that are readable. So now the question is, how do we evaluate this, or how do we make these decisions? And that's what we're working on now. So right now I have run 200 specimens through the entire pipeline. We have many more in the first step, but in this last few steps, I've run them many times as we start looking at these metrics and trying to ascertain what works well and what doesn't. And about 80% pass all those computational tests and produce really, really good labels. But that's as determined by me, by eye. When they passed the test and I went and looked at them and went, oh yeah, those look pretty good. Just under 3% failed. And so far, all of them were butterflies that had significant occlusion. And then this one anomaly where it's trying to push a round peg into a rectangular hole to make that label a rectangle. And I hadn't really ever thought that there was going to be round labels, and no one told me there could be round labels. So we don't actually have a tool in place for round labels. So it will fail on a round label because I don't know how to make that a rectangle. I mean, there is a mathematical mapping, but we haven't looked at that. And then there are a bunch that say maybe. And I'll talk a little bit on the next few slides about what maybe means, because not all, the ones that fall into that category are often pretty good. So here's an example that fell into that category of, well, it didn't really work. It might need exception handling. And what happened is there was significant occlusion on enough views of the two labels that it's got some erroneous stuff there. And we think we have a process in place that can handle that. But for the most part, if you look at these, they're pretty readable labels, and you can extract the relevant text information. Here's another example that fell into that category. And actually, they're all pretty good. There's just a little bit of an imperfection here in the alignment. But again, we think that those are probably, so of that 15% that fell into this, we don't, maybe category, we think a significant number of them are going to be readable. We're still working out how we'll handle those cases. And so where do the problems occur? As I said, occlusion is a problem. When there's a lot of labels or when the specimen overlaps the label, that's usually where there's a problem. In some of the samples, the labels are very, very curvy. They're very bent, and you get some significant warping. So those are the two things that I've seen. There are a few handfuls where Mark talked about our process of using M words and matching words, but we have cases where the labels have identical words on them. And so then that process fails because you don't know which label, which word, is this on that label or is this on this label when you look at those words. So there are a few cases that because of the assumptions we've made, it's not going to work. But so what we've been doing right now is, again, going back to that best in breed text detection, taking the virtual labels, running them through it. The top example works perfectly, gets all of the text. This example, it's missing some characters there because we didn't get a view where we could remove the specimen. So there's a few characters missing there. But so far it's looking fairly good. Again, some small text and, didn't read the word tall there. It seemed to have problem with the degree sign, and we actually think we know what's happening there. And it probably didn't have a problem with the degree sign. That's more our conversion to printing it on the label that had the problem. But we're working on this. This has been very tedious because I've been manually looking at, did it correctly do it? But we have, our collaborator has manually transcribed verbatim 200 specimens. So we actually have a gold standard test, and we're going to be using that over the next couple of months to make this process more rigorous so that we can make that final decision of, what is the approach that's going to give us the best labels. I'm going to turn it back to Mark for the last little wrap up.
So just a couple of closing words about kind of what the man behind the curtain is all up to. So this is about the infrastructure of the pipeline itself. Just a couple of simple remarks. First of all, we've already told you that we've got this process in place where we develop the algorithms necessary at each of the stages. test them, try to find out what's working, what's not working, settle on something, and then we go through and we parallelize it. We take it and kit it out to be able to run on many, many machines. We like to work on our laptops for the most part, as everybody does, but we ultimately have to use more power to go through lots and lots of images. And so we are making some serious strides into getting this kind of job done. We're up to 2,000 specimens of processing on a fairly big hunk of the pipeline, but that's out of, as you see, as of about a couple of days ago, we've got this steady influx of data coming. 200,000 images as of a couple of days ago, sort of folded into almost 7,000. So we are at least working at the scale of the data that we're collecting, but it's going to get away from us if we can't bring it all home. And then this is just to sort of summarize the kind of tidbits. Over there is a picture that shows, is meant to imply that we've got just buckets of little data pieces that we're using. Out of Nicola's pipeline, we end up with fragments, individual fragments, composited fragments from all of the specimens that she does. And so we have lots and lots of data to be playing with. And of course, the first thing we have to do is finish doing the parallelization of the pieces that fall out of her workspace and bring them up to the point where we're doing thousands and thousands at a time. And the second two bullets just summarize where we are with that. You've heard that story. And then finally, our next bits of work are going to be closing the loop on the choices made. And that's going to involve making some critical comparisons with the ground truth data that Zoe Flores provided to us of a fairly substantial number of images taken from the instrument that she has access to at Harvard, which is the same COPUS system that we're using here at Yale. So with that, I think we're done.
Very much. Okay, next we have Hugo Reyes-Centeno. Hope I said that correctly, from the University of Kentucky. And for those of you who are joining us virtually, if you look on the agenda, we will have breaks for questions after some groups of talks. So after this presentation, we will have a break for question and answer. And if you want to put your question in the Q&A for now, you're welcome to. Second, thank you so much.
Very good morning. And great presentation to start us off in the day. So I'm going to step out a little bit and give you a more general overview. I primarily want to share with you some new resources that we have at the University of Kentucky along the same vein of the kind of project we just saw. But the emphasis really is on multimodal imaging, meaning that we have a diverse way of capturing data, imaging data from a very large environmental scale and down to the kind of artifactual level that's represented in the previous presentation.
Okay, you're good to.
And this is stemming from a project that is funded by the National Science Foundation on heritage science. And the term is very common in Europe, and I want to bring it up here because here in the US, we're not as familiar with it. But the United Nations, for example, defines heritage in this way as something that we live with today from the past and what we value for future generations. And so while this cohort is primarily interested in the natural aspects of heritage, I as an anthropologist and my colleagues at Adu's lab are also interested in the cultural aspects of that. And so we really are looking at both aspects of inheritance from the biological and cultural side. And as we've already seen, and as this group is very familiar with, drawing from established disciplines to try to get at some of the questions we're interested in, some of the more challenging problems. And underlying all of that, and what brings us here together, is that we're using different mathematical and computational approaches to do that. So in Europe, there's a wide infrastructure for heritage science, and it's something that's been developed for a very long time. For example, in 2005 at the government level in the United Kingdom, it was recognized that it should be a priority and that there was increasing opportunities. And one of the challenges here in the US is that there isn't quite a unified approach. iDigBio is a fantastic example of trying to get to that. And there is very much demand So my background in anthropology and archaeology, we're out in the field, we're collecting data, and oftentimes this ends up in repositories of data where it's just no longer being used. And because of the economic infrastructure revitalization efforts that we've had, there's increasing data being generated. So as an industry, there's been consistent growth, projected growth into the future as well. More data will be generated, and a lot of it, unfortunately, will probably be sitting on shelves for a long time. And so that's something that we want to address at Aduce Lab. And so what we did was to apply for National Science Foundation funding through this solicitation scheme called the mid-scale. And our mid-scale is on infrastructure for what we then call heritage science. So if you're not familiar with it, just very briefly, mid-scale is meant to be this bridging gap between these very large infrastructure like the Argonne Laboratory, like Lawrence Laboratory at Berkeley, and then those smaller laboratories, individual PIs and so forth. And so we're really bridging that gap. And in this case, for questions of heritage science. We call it Aduce because, of course, we're trying to bring something out from data and something that is hidden or latent. And one of the best examples is from the work of my colleague, Brent Seals. And so you've seen examples in the previous presentation of text that is at the surface, difficult to get because you have overlapping text from this needle and we don't want to move that. But you might have some cases like this where you have a scroll that is completely burned and physically we cannot unroll it. We try photogrammetry as in the previous examples, but we're not going to read anything because it's all inside. So what do? Well, one way we can try to get at this, and this has been really more than 20, 30 years of work in developing different techniques, but is to look inside in a non-destructive way using micro-CT, microcomputed tomography. And here you can appreciate just from this image how it has been warped, but how you can still see the layering of the parchment paper. And so the question is, can we actually detect the ink on this text? And well, sure enough, in this case, one of the Dead Sea Scrolls from Engedi, after a lot of work, you can get at that text. This was a relatively easy, thinking back at the process example, because the ink that was used for it was lead. And so you put that under micro-CT, there's a very high contrast between the parchment and the lead. There are many more challenging examples, particularly the scrolls from Herculaneum in Italy, where the ink that was being used is carbon-based on papyrus paper. And so the level of contrast between the two with CT is actually not going to be very high. It's going to be high if you use infrared, for example, as another form of imaging. Then you're limited to just the surface of some of these scrolls. So what do? Well, we can create an AI model where we use some of the infrared data as part of our training data set. And we can also use data from experimental context. You know, we have our students write on papyrus with different kinds of ink, different compositional elements, and then go on and CT scan that and create a training data set that can then be subjected to a neural network model to get at that hidden ink in a micro-CT case. And so there's been great advancement on that and great success as well. Other forms of imaging that might help us out, that have helped us out, is through scanning electron microscopy, just to understand how ink is being absorbed by the parchment, and again, using experimental data for that as well. And there are, of course, easier cases, as is the case here, where you may not have to completely unroll a scroll, but you do have to get at these different layers. And so this is one example of what we're developing at Aduce Lab. Traditionally, as I mentioned, it has not been very easy here in the US to do that. We've had to rely on laboratories, as you see in the description here, that a dentist was going to do this work. Well, it was a dentist because we were using dental equipment, but now we have a dedicated facility at University of Kentucky where we can develop this. This is different from what I was used to. I did my PhD at University of Tübingen and research at the Museum of Natural History in Paris. So there we had dedicated equipment as well. Here you see a Neanderthal skull and a CT scanner being scanned. And the kinds of questions we're interested in, this is more my line of research, are trying to get at the different morphological aspects of some of these extinct humans like Neanderthals in a non-destructive way. And so looking at the inside of skeletal material, we can get at some of the features like the inner ear. And, you know, part of this process would be doing the multimodal imaging first, photogrammetry, looking at the outer surface, getting information on color, and then doing micro CT scanning to look inside. And it turns out that we also do this because we sometimes want to sample some of these specimens, whether they be human or otherwise, for molecular studies, for ancient DNA. And in this case, it actually has been shown that the temporal bone and the petrous portion of the temporal bone yields very high quality DNA. And so prior to that kind of destructive analysis, we want to get the best kind of imaging possible and leave a record for that. And I'm sure you've seen the kind of inner ear reconstructions that we can produce with micro CT. And, you know, this kind of work is very promising, very multidisciplinary. We're very proud in the anthropology field that we have a Nobel laureate now in 2002 for doing the kind of work that was in sequencing ancient DNA. But of course, there's a lot that went in through that development, including the conservation aspect and the imaging aspect involved in that process. So this is our group, multidisciplinary, and many of our other colleagues that are involved, primarily engineers, as I mentioned, because traditionally we require a lot of that mathematical and computational background, although we're starting to develop anthropologists that have this kind of background as well. And we will be taking on various projects into the future. So I'll just go through briefly how we're structured. There's a bench component, which is a lot of the kinds of equipment that you are used to. And, you know, depending on the research problem, we might actually not use all of the equipment here, but in many cases, we want to maximize the kind of data that we collect. From the two previous examples that you've seen, it involved the imaging through micro CT, it involved optical imaging of the surface, and it involved scanning electron microscopy. So we can create this pipeline to generate data that can then go into some of these models, AI models, machine learning models. And ultimately, we want to make our facility as flexible as possible. And so, as in the previous example where there was machinery involved to conduct photogrammetry in a very specific setting, we want to develop that kind of setting as well. And this is part of the Flex working group. And one of the other components that we're very proud of and that we're very excited about is the co-design. And so, of course, as scientists, we are interested in specific questions, but involving the community in that co-design process and understanding what the broader general public might contribute to our approach. And then for me, as an archaeologist, I'm out in the field a lot of the time. It really starts with collecting the data, the original data, primary data, often through excavations in this case. And so we have a mobile component where we can take some of this equipment and go out in the field and collect imaging data at this broad scale, including photogrammetry with drones, but also underneath through ground penetrating radar. So some examples, this is ongoing right now in Rhode Island at the Buttsville Fort in Portsmouth, have students now collecting data. There's been LiDAR data collected at a broad scale, and now they're collecting ground penetrating data at a smaller scale. And of course, that co-design component, having the community involved is important. And then the cyber component, simply, you know, being able to collect, store, and then process a lot of this data. So where we are now is that we, in terms of in the view of the NSF, are testing a lot of this equipment, the synergy between the different components. We'll create policies and procedures, and then we will officially be commissioned in autumn 2026. And I look forward to a lot of collaborations with this group here. So thank you to the organizers, and thank you for your attention as well.
So we have a few minutes for questions. Yes.
I have a question for Hugo. So when you have the TEMs and the SEMs, do you have special fixtures to be able to help do correlative work? Or is this something that you're planning, or, you know, what you have lots of different length scales there? How are you going to bridge those length scales?
Yes, yeah, so this is part of what we're testing at the moment. Of course, with every artifact or with every kind of sample, it's gonna be
Unique.
different. Yeah, it's gonna be
Okay.
unique, and that's really one of the challenges that we have. When we, for example, to give you a good case scenario, when we had the data of the Engedi, we thought, oh, we could apply this to any scroll that is burned. And then we went to Herculaneum, and there's just no contrast between the ink there and so forth. So yeah, it's gonna be unique to a lot of these different questions, but it is an advantage to have a centralized place to do this and to test it.
I have a stupid question. When you're doing the imaging of the invertebrate tags, you keep them lined up one on top of each other? And if so, why do you not fan them out so you can see them from different angles, or because it seemed like.
So we don't want to disturb the specimen on the pin.
Sure.
So someone has to actually take it and put it on a fixture. Actually, we have an expert here who does some of the imaging here at Yale, but you could do all sorts of stuff. I mean, in Finland, they take them off the pin.
Right.
And that makes the task a lot easier, but it takes time and it risks damage. And so
But you have to remove, you're removing it from its original bedding to move it
sticking
to
it on,
the imaging
but
place anyway.
just one motion and then back.
Okay.
And so, you know, you could spend a lot more time and image a lot more carefully, but that's really time consuming. So the idea is you just take these images and then pull the data, and, you know, it isn't that in view, you know, pose two, you always see the labels. I mean, it's in one, you know, the probability is that you're gonna get the labels over the multiple poses, we hope.
Is there a kind of success rate increased with amount of time processed? You do know what I mean? Like the more time you spend fanning out the tags, the multiple tags, the success rate for all those.
We don't do that. We don't do
You
that.
don't touch
We
it.
don't wanna touch the
The
stuff.
only two carbuncles that I can think of that are kind of wrinkles on just picking up the thing and putting it in and putting them back. Is that one, sometimes in some collections, the labels are too close.
Right.
And so there would have to be some sort of a, you know, touching and sort of separating the
Yes.
thing. The other carbuncle, I can't remember right now. Oh, yeah. Many collections want to use that opportunity of touching the specimen to put on a QR code that
Right.
might not be there.
I see.
And so that means that you have to somehow include in the process without adding much time to the workflow. But our whole gig is zero time or little time, because I would love to get to kind of 1 second per insect.
Sure.
And that way you could do kind of, you know, million a year, and then you'd be at the point where in our lifetimes, we
Imaging
the whole
and the
collection.
trays themselves, you know, not have to take.
There's a lot of problems with that, not least of which is that NSF hates the word drawer at this point. They are really, I think there's a little bit of a virus that has yet to clear itself.
Hey, Mark, and Nicola, on the label positioning, like, I see the point where like if they're directly like touching each other, that's not great. But there has to be some
Yeah.
distance cleared, right? Like, what, do you know what that distance is where like you can see enough of the context of a label to like
Well, so
recognize where like
we've
that.
done some calculations, but it also depends on how far, and, you know, we're trying to keep the cameras far enough away that we're not ever risk of colliding, but you can do that calculation. We've done it.
Yeah, the real answer will be, as we take a bunch of data, we'll kind of we'll have some emergent statistics that will tell us. We went into the process though with a model that we tested where we put pseudo labels on various pins at various angles and various sizes, and asked the question, what is the best sort of elevation angle for the cameras that would see furthest into for various separations? And we came down to the notion that we could probably handle as little as 2 or 3 millimeters separation if we chose angles that were appropriate to that for a typical range of widths of plates. But it's all statistical in nature. And so the whole process in the end has to have a thing that says, I got the answer or I didn't get the answer, so that you can kick things out to sort of human in the loop or reconfigure for some special
And
cases.
the optics matter. So the first pass that we were experimenting with, we had no budget. And so we used crappy cameras, and then, we got this grant from NSF that, Nelson bought really nice cameras. And so it made a huge difference to have the high quality optics. I mean, our success rate just skyrocketed because we have much better cameras now.
A lot of parameters to juggle.
Yeah.
Okay, we're gonna need to move on to our next speaker, Peter Cerda from University of Michigan. So much.
Hi, thank you all. So I'm going to talk a little bit about how we're preserving and sharing museum CT scans at an institutional data repository. So I'm Peter Cerda. I'm an associate librarian and data curation specialist at the University of Michigan at our Deep Blue data repository. I also did my PhD in the Museum of Zoology at Michigan, so I was really happy to learn that I was going to keep on working with this group.
Sorry, I'm gonna.
Yeah, no worries.
Sorry, technical difficulties. Okay, I don't know why it's doing that, but just keep going.
I can read it. Okay, so at Deep Blue Data, our ultimate goal is to help researchers share their scholarship that they produced at U of M. So our areas of focus are really helping folks deposit their work, making sure that we can that they can be discovered by folks looking for their research, and that they can access it. So they can actually download the data sets, reuse it for their own work. We provide a curation service, so we help the depositors try to achieve these goals. And ultimately, we preserve the work for up to 10 years, probably longer. So on our end, what we're actually helping researchers do is describe the data. So work with them to, improve their documentation. So like how many of y'all have downloaded a supplemental material, opened a folder or file and had no idea what you're looking at? Right, we're trying to help them not do that. And we're also trying to improve their metadata. So that way search engines can pick up the data set so you can actually find it, right? And all of this is really helping, or all of these are moving towards the goals of trying to develop fair data. So by fair, I mean findable, accessible, interoperable, and reusable, ultimately. So we need really good documentation and metadata to really achieve these goals. So in the end, Deep Blue Data, we host the raw process data, code, media, et cetera, and it's all openly available to y'all. Right now, we're at about 1,200 data sets, and with the addition of the CT work at the Museum of Zoology, we will probably break 2,000 by the end of the year. So we have very good relationships with the Museum of Paleontology and Museum of Zoology. I'm going to go through our relationships with both of them, but the paleontology collection is probably the easiest one for me personally because it's self-deposit. So they just have to upload the data set. Usually it's Adam Browntree that's doing that. And we have basically worked with them to a point where they have pre-curated data sets. So we already worked with them on the metadata. And there's very few things that they actually have to do before they upload it. If they have a big data deposit, which is usually more than 5 gigabytes, then they'll contact me. We'll get it up there. And it's about 40 data sets at this point. One interesting thing that Adam actually told me is that the inclusion of a thumbnail in their deposit actually really helps increase the usage and finding of it. So that's something we advise to all of our users is to include a thumbnail, because who doesn't like looking at CT scans? So the problem child is the Museum of Zoology. So because now they've established a core there, they are popping out CT scans like crazy. So this is a little bit more involved, just to keep up with the scale that they're producing at. So this is more of a facilitated deposit where I work with Ramon and Cody at the Museum of Zoology to upload all their data sets. And my predecessor actually created a set of custom scripts that I'll go through shortly to kind of create the deposit so it's not too much work for them and more work for me. And at this point, we have about 170 data sets with a lot more to come. So as I walk through this ingest cycle, basically the one big time constraint is actually moving the data set from their server to ours. Like we're talking about data that's in the hundreds of gigabytes. And thankfully we're on the same network, so it doesn't take too long, but it could still take half a day to move a large tar file. Once we actually have that, then I'll unzip it. That takes some more time. And then we run an organization script that basically sorts the two different types of scans that we have, or the data that they're providing. They have the raw CT data that they're giving us, as well as the reconstructed, the derived data set. Then I run this heavy lifting script, the UMMZ GBIF script, which basically pulls it asks GBIF for the record for that actual data set or that individual specimen. So we can pull the taxonomy from it, a bunch of the metadata that's already up on GBIF, pull that and associate it with that record in a YAML file. And then it also tars the CT file so we can actually upload it. Then we upload it to Deep Blue. This is where I get a little bit more involved with our system and get it up there. And then we try to upload the metadata to MorphoSource. And this is actually the area that I'm working on now. Now that we got this really up and running, trying to get the metadata up on MorphoSource so they can act as an additional discovery layer. So more people can find these data sets. All right. And then just a quick acknowledgement. I also have to thank Adam, Cody, and Thompson, and Ramon at the different museums, as well as Susan Borda, who really developed a lot of the scripts that I'm using, and Fritz, our developer. And then if you want to take a look at the scripts, you can find them at this website. Thank you.
Rebecca Dikow is up next from the Yale Library. Thank you.
Everyone. For my notes, so I hopefully stick to time here. My name is Rebecca Dikow. I'm brand new to Yale Library. I've only been here, gosh, I think it's 7 months, but it feels like 2 weeks. I was previously at the Smithsonian Institution, and I'm going to talk about this work was done at the Smithsonian. This paper was published in a special issue of the International Journal of Plant Sciences that was a tribute issue to Vicki Funk, a botanist at the Smithsonian. I think it's because it's a PDF, probably.
Maybe.
No worries. This project was inspired by work done by Vicki and also Effie Capsalis, both of whom we lost much too soon. As part of the American Women's History Initiative, which is a digital component of what's going to be the Smithsonian Women's History Museum, which will be opening physically, I don't know, maybe 10 years or so. But through this work, part of this was accumulating a list of women who worked at the Smithsonian, which is more difficult than it may seem, because most of the women who worked early on at the Smithsonian weren't employees. They were spouses, they were volunteers. It just obviously, we all know women weren't employed to do science regularly. So right now this list has more than 500 people on it, which is really exciting. And just to sort of illustrate sort of how we came to this work, Mary Vox Walcott is one of the women on this list, a very early Smithsonian, not a staff member, but a spouse of previous head of the Smithsonian, Charles C. Walcott, who found the Burgess Shale, very well-known paleontologist. But Mary in her own right was a botanist and a naturalist, a painter. And you can see a specimen she collected and the painting. So she published a lot of her work. Here are two specimens. This part might be hard to see here. But in 2019, we had an intern on my team who found specimens. In doing this work to sort of understand more about women in science, she was looking at all of Mary's specimens, but there were some that were attributed to her husband, but were collected after his death. And I don't think you can see that label here, but I think we zoom in here. Maybe you can see these are collected on the same trip, July 1937. On the left, you see in the bottom, Mary Vox Walcott, and on the right, it's Mrs. Charles D. Walcott. Okay, it's a classic problem. We weren't the first to figure it out, but it's like, okay, how often is this happening and what can we do? Here's another interesting specimen with the same problem. And this one is just, it's sort of comical because Mary Walcott was friends with Lou Hoover, who was the first lady. And they did lots of fun stuff together, collected plants. And you can see this specimen is in the database. I think it's been fixed, we can hope, as Charles Walcott and Herbert Hoover. And it was really collected by their wives. So, these are very well-known women. There's so many other women who, have been really just lost to history in this way. So we were thinking about the Funk List and what could we do to sort of look at how many women at the Smithsonian this had happened to and what we can do to do this work. So we used Bionomia heavily in this project. Here's just a screenshot of Vicky's page in Bionomia and all of the specimens she's collected, just an amazing career. I think she collected almost 300 families, 270 families or something, just huge, amazing. And so we focused initially on women who were deceased. There's challenges with sharing privacy issues. So we looked at all of the women who were deceased on the Funk List, and we were able to attribute specimens to 40 different women, which is a lot, but also not a lot at the same time. And so we'll talk about sort of the challenges there, and more than 120,000 specimens. So here's the number of specimens per both on the left is the collections, and on the right, I think, are identification. Sorry, it's a little bit hard to read. So, and this is also sort of by department. So many women collected and identified not only for the Smithsonian, but for other institutions. That's also kind of an important way that Bionomia can really add to the sort of legacy of people's careers. So it's not just the work they did for their own institution, it's sort of more broadly. These four women in particular, Mary Agnes Chase, Mary Jane Rathbun, Doris Cochran, and Doris Holmes Blake, very early Smithsonian workers, collected specimens for at least 4 departments at the Museum of Natural History, which is just really exciting to see. We also mapped the data onto the Open Tree of Life to look at, you may think, well, the Smithsonian has, 157 million objects or so. We're only talking about 120,000 specimens in this work, but if you map it across the Tree of Life, you really get amazing coverage. And I think that's just sort of highlights the impact that these women have had. It may not always be in numbers, but it is really just remarkable. We also mapped, so I encourage you, I can share the link to the paper so you can zoom in a little bit. It's hard to see here. These are the kind of our top 4 botanists, and just showing the difference in terms of how much collecting they did and how much identifying. And you do, we haven't done like statistics on this, but you do notice a trend that earlier on it was much more in terms of identifications, and it's sort of kind of just, women's ability to travel was also obviously limited for a variety of reasons in the past. And these are the top 4 invertebrate zoologists, a similar pattern here. And really we found botany and invertebrate zoology to just have the most women and the most collections from women. And that's for a number of reasons. It could be, there were more women working in those departments. Also the digitization is more complete. But yes, entomology, invertebrate zoology, there just has to be a lot more work done for digitization. So some of the challenges we identified is, as I mentioned earlier, is that titles, Mrs., Miss, present on paper labels are often not included in the transcribed data. So this right away, women are often referred to as their spouse's name. And so the specimens get attributed to their spouse. So the recommendation is to look at images. How we did, we sort of went to the image and looked, sort of identified this problem. We could, potentially use regular expressions to search for multiple name permutations, which we did as part of this project. Also, collector and identifier names are written inconsistently, right? Walcott, there's C. Walcott, C.D. Walcott, Charles Doolittle Walcott. Again, using identifiers, unique identifiers is the way around this problem. It's just a huge amount of work needs to be done there. Another thing we noted was that when dates sent to GBIF don't have a month and year specified or have a 0 for either one, the dates can be rejected. So we had to do a lot of manual work to look for those specimens. Obviously, these recommendations are pretty obvious, but there's just so much manual work that needs to be done, unfortunately, at this point. Dates are really important for this question because it can really help us know if this specimen was actually collected by one person or another. In entomology, we found challenges around specimens that are part of a series. They may not all have labels. So then when they get digitized, you don't necessarily have the name of the identifier in the database. And so those specimens don't get attributed to anyone. So the order of the specimens matters in digitization. And so it's sort of that we need some better workflows to make sure that people get credited with their work. At least at the Smithsonian Natural History Museum, the type specimens don't consistently include species authors or under-identified by. So we're really missing this. One example is Doris Holmes Blake, who I think described 800 species. She's an entomologist, and she's very, very poorly represented in the database, even though we know that she did all of this amazing work. So in this case, we're checking other fields where specimens are, if we know their holotypes, and add the identifier, the person who described the species to the identified by field. The biggest challenge is not all collections are digitized. So we are using other documents, things like expedition and travel logs to gather information about collectors and researchers' work. I think particularly for people who are, weren't employees, this can be really helpful to know who else went on a collecting trip. And we can really highlight their work. And I'd like to thank a lot of colleagues at the Smithsonian and the Because of Her Story project. We funded this work.
Winchester will be up next for MorphoSource.
One more thing, and if not, we'll just keep going, but I think we're gonna fix it.
That's good.
Yeah, so while she's doing that, I'm Julie Winchester. I am in the Evolutionary Anthropology Department at Duke University, and I'm the technical director for MorphoSource, which so MorphoSource got mentioned in Peter's talk about, the work at Michigan, which we're really excited for that.
Yeah,
sorry. Roll with it like it is.
Roll with it,
All right.
sorry.
And this is MorphoSource, a repository for ants.
It looks good online.
That's the important part.
Full screen
There
online.
we go. Well, with this, we're here for digital stuff, right? All right. So I'm going to talk a little bit about MorphoSource, say, just to give you my real quick elevator pitch for what is MorphoSource. MorphoSource is a digital repository service open to the public where researchers, museum curators, and the general public can find, download, and upload 3D, but also 2D and AV media representing physical objects of academic or scholarly interest. So so it's mostly physical. There are some born digital, and most of those physical objects are natural history specimens, but some of them are cultural heritage objects as well. So just to give you a little bit more info, free digital repository at morphosource.org. We are 3D specialized, but we do allow and support 2D and AV media. And the data are all contributed and managed by our user community. We don't upload the data. We don't own the data. We're just helping users make them accessible. We mainly work with researchers, museums, and imaging facilities in the general public. And we support data discovery, data access through downloads or through our IIIF-backed web viewer with previews, which tomorrow I'm going to talk a little bit about IIIF as well, allowing people to contribute and publish data, and also helping museums and now imaging facilities to manage and track scans representing their collection or imaged with devices from their facility. Just some basic facts and figures. The numbers on the right, they're a few weeks out of date. I think we are now at about 197.5 thousand media data sets, where each of those data sets is either an entire CT scan or a 3D model or maybe a 2D slide collection. And those represent almost 70,000 physical objects. We've been going for a little more than 10 years. We are headquartered at Duke, but we are financially supported by generous help from NSF, some Duke commitments, and by data contributors in some cases, usually data contributors that have NSF and other public funding that gives them some funding for data storage. And we work with a lot of different groups, a lot of working groups. We work with IDBio, GBIF, IIIF, to really try and advance things in a very standards, community-focused kind of way. That's why I titled this that we are a community repository. Just to show you some things, these are the types of media that people put in MorphoSource, your 3D meshes, various image stacks, so that could be CT, but it can also be image collections for photogrammetry, or histological slide samples. We now support that. Various 2D images alone and videos. And I don't expect the people in this room to read this, but this is the big list of 14 imaging modalities that are supported. If you can read this, everything that is underlined, those are ones where we have some modality specific metadata. We're actually totally open to adding better modality specific metadata for the others. It just sort of takes a community individual or a group to come and be like, this is what my community really needs for RTI or some laser aided profiling or something like that. So one of the things that MorphoSource really focuses on is we have a strong focus on allowing users to document workflows that produce images. So, when you make 3D images, when you make CT images, there is usually a very long workflow. We've seen some of those workflows today, sort of the garden path of how you produce these things. And MorphoSource's emphasis is that we have a data model, we have a data vocabulary, which the data model and data vocabulary are all published in machine readable formats for allowing users to document and curate that whole process. So this slide's a little messed up, but I'll walk you through it real quick. That say if we have an institution that maybe manages both a physical object and a device, all three of those things can have records in MorphoSource where they each have their own metadata describing them. And then we start to relate things through events. Say maybe a device is used to image an object in an imaging event, which has its own imaging event specific metadata to produce that first media. And then, it's maybe a CT stack, but your CT stack may have processing event that produces derivative media. So you can build these parent-child processing event chains to curate and document in the site, not just, hey, I have a mesh, I'm telling you it represents this, but hey, I have a mesh. I made it by smoothing this other mesh, that I made by cropping this other mesh, that I got from segmenting it out from a CT scan, that I got from an object. So you have very strong linkages between those things. And we think that's what ensures that data remains data, is the context, basically. Yeah, and one aspect of this is that MorphoSource is built on a museum-mediated flow of data. So for instance, all of the media that represent objects, they have to be linked to a physical object record. If a museum is publishing their records through iDigBio or GBIF, we can then link the object record in MorphoSource to the aggregator record. So you don't even necessarily have to fill in much of that metadata. We can automatically pull it in from iDigBio. We can update it as time goes on. And so again, strong linkage from the museum database to the media in MorphoSource. And because of that, because we have that linkage, and because we have the linkage to also devices, which can be associated with facilities, that means that we can empower museum collection and imaging facility staff. So you can make, if you are a staff member at an institution like that, you can make an account on MorphoSource and you can gain elevated access to see what's in the site that represents either your collection objects or things that were scanned with your imaging devices, to track them. And for object collections, we actually have some features to allow you to, if you choose, participate in sort of long-term data management of media automatically. Yeah, and just to point out, the site does see active use. There's our downloads on the left, views on the right. In the downloads, you can see there was definitely a COVID spike there. The date axis is messed up. This is right now this is actually 2016 to 2024. And this is 2021 to present. So I think something when it maybe Google slides, it's fine. Just really to point out, this is the pandemic, unsurprisingly. But we have had a recent bump, which I think is in large part related to the OVERT project and them publishing the big fancy final OVERT paper. So we got a lot of publicity from that. Though I will say, OVERT's phenomenal. It's incredible. I think Ed's going to talk some about it. But I will say it makes up actually less than 10% of the media in MorphoSource. So, yeah. Repository breakdown. We have about 1,000 cultural heritage objects. We are working to increase that amount. It's mostly biological specimens that hail from 3 kingdoms, 8 animal phyla. I feel like fish and herps are always warring to be the most numerous thing on MorphoSource, but also strong representation from mammals, inverts, plants, birds, some dinosaurs. Just to show you some things. Yeah, so we do have insects. So insect photogrammetry there. I believe, hopefully, Yale's going to be adding a bunch of new insects. We have flowers, echinoderms, and other cool inverts, protozoa, and yes, art and cultural heritage objects. All right, yeah. So I think I talked through some of this stuff, various things you can do on the repository. For user accounts, you don't need an account to search or browse, but you do if you want to download media or to access private media. You can also apply for contributor status to upload media. Yeah. When you are contributing, you can contribute single media one at a time through a decision tree, like a user-friendly. It says, hey, what kind of data do you have? I'm going to send you down the right path. I'm going to then ask you a question. For better or for worse, our inspiration for this was TurboTax. It's like a TurboTax approach to trying to teach you what you need to know to upload all this media. Yeah, there's explanations for each step. The process is different for different types of media, whether it's derived. But contributors can also batch submit multiple media at once. Right now, you would do that by uploading files to an SFTP server, but we're actually going to shift over to Globus for that. And then you upload a metadata spreadsheet to a web UI. Yeah, data viewing, you can see a lot of cool stuff on that page. So if you go to a media page, it'll show you obviously the media with a preview that, as I said, that's actually IIIF backed. There's IIIF manifest on that. So you could take that preview and embed it in other websites. You could even use it in other viewers. File metadata, things about the object. image acquisition and processing details. We mint identifiers. So we mint an ARC for everything in the system, but we can mint DOIs when users request them. Sort of all the other good stuff, ownership and licensing. Yeah. If you download things, you can download individual media, or you can group them together in a media cart and download things all at once. When you download things, you see a download intent survey that'll ask you basically, what's your usage of this? The reason why we do that is because this gives data contributors the ability to track the impact of their stuff. It's about, and it allows for the creation of sort of an alternate currency of scholarly impact. So you also have to agree to terms and conditions. We have a download use agreement. It's real simple. It's pretty much just, hey, you have to attribute us and the object institution if you're going to use this stuff. But contributors can customize their usage agreement. And if you're a museum, you can actually supply like your collection's standard usage agreement, and users have to explicitly say, I agree to follow this before downloading the media. Yeah, there are a few different controls of how media can be set up in the system. You can set them as private to where only you can see it, or open download where anyone can download it if they fill out that usage survey. But you can also set your media as published with restricted download, which if you set that, other people can see it, but to download it, they have to send you a request in the system. They tell you what they're going to do with it, and you can choose whether to communicate further with them, to approve the usage, in which case they can automatically download it, or to deny it, because in some cases that is necessary. Yeah, there are lots of different search tools on the site. I say search, explore, and filtering, because sometimes people don't realize this. If you go to MorphoSource search and you search with an empty query, it'll show you everything in the site you have access to. And then you can use a whole bunch of different filters to filter by media type, the type of object, the organization that holds it, modality, all kinds of different things. All right. Media can be grouped in projects, projects are, I own this media, I want to maybe share it with someone, but we recently added media lists. They're basically like playlists. They're like YouTube or Spotify playlists. Any user on the site can make a media list, and any user can add any published media to that list. So like I have a published list up there right now called platypus scans. I just went through like all the platypus scans I could find on the site in there. So you can use that for lesson plan collections, themed collections, all that kind of stuff. All right, the last thing I will talk about is we have, as I've been mentioning, that we have these museum collections or image facility records. So we group those under what's called an organization record. That's any like bureaucratic autonomous unit, because sometimes an entire institution will manage all of their stuff in one group, or they'll split it up into 12 different divisions or 18 different collections. But organization also applies to imaging facilities. And then media get associated with these object collections through objects, and imaging facilities through imaging devices and events. If you are a collection staff member, you can manage your organization on MorphoSource. It unlocks a bunch of different features, as I said, including the ability to view and track all media associated with your collection or facility. Yeah. Object collections can additionally provide some recommended ownership and license settings, and they can choose to serve as the long-term data manager of published media. If your museum or your collection does this, basically when someone deposits data, they get a little pop-up that says, this museum seeks to exert long-term data management control over this media. You can either elect to have that collection make the decision now or when the media is published. So you can't embargo media, but we do think that specimen collections tend to be one of the best long-term stakeholders. We're thinking about media management 100 years down the line, not necessarily 2 years down the line. So museums can participate in that process, but also there's a lot of flexibility. If there are situations where the researcher needs to be the one to hold management, the collection is completely in their power to say, sure, go ahead. Yeah, if you have already been using MorphoSource, you probably received an email, we made some changes. I never mentioned the word team in this because we got rid of that concept. We just simplified it down. And your organization now can actually manage directly. Yeah, oops, one last thing. That's fine. Just that imaging facilities, we just recently added the ability for imaging facility managers to manage and track media. Basically, you can add devices, edit devices, and, oh, I see. Yeah, you can track the media, you can add and edit devices, and you can see usage statistics. All right. Thank you very much.
Ah. It wasn't my
Maybe it's
computer.
a slide in the thing. Yeah.
There's a lot of computers up here.
So we have 15 minutes for questions. I do see there is one question. Summer, do you mind reading the question in the Q&A for us, please?
Yeah, this is for Julie from Nikki Nicholson. Given its funding, is MorphoSource only available to support US researchers? And if so, is there an international equivalent?
MorphoSource is intended to support a global community. I mean, that's pretty much we have, I don't remember the numbers off the top of my head, but we have representation from many different museum organizations around the world. We have many contributors from around the world. I will say we've been in talks with some colleagues in the UK for a project, actually a couple of different projects in the UK that might eventually see a sort of a federated node of MorphoSource spun off, across the pond, maybe in the UK or elsewhere. But that's really more just about that wouldn't establish two separate repositories. It would more just establish two different nodes that would see data in each other and could maybe be targeted at different groups. But no, we're globally focused. And yeah, we welcome contributors and data users from anywhere.
Yes.
I have a question for just Rebecca. You had said that sometimes the digitized version, there just isn't digitized data that you can draw on. And when that happens, you turn to records that are logs of trips and things like this. Are those digitized?
To some degree, there's been a number of projects, Field Books Project, some of VHL's work, travel logs, and not everything by any means, but the Smithsonian Archives has the personal papers of a lot of these women as well, so we can sometimes go and actually look at the physical objects, but they're not.
Do you have a process where you can say, oh, I like that, that would be very useful, and then point to it and have Smithsonian go and digitize it and add it to?
I have it. I don't know if it's a formalized process, but.
Yes, Steve.
Yeah, so this is for Julie and Peter both, I guess. I think you both mentioned that you pulled metadata from GBIF to fill in taxonomic information and so on. I'm just wondering, do you have a part of your workflow to round trip your media information back into the GBIF record?
Yeah. I'll let Peter.
Not at this point. Something we've been thinking a lot about at Deep Blue is how to link our data with its other corresponding other data sets or the linked data. But we haven't been able to incorporate that yet. That's something to definitely look into.
Yeah, so I'll say with MorphoSource, there was a period of time, I think back in the dark ages before the pandemic, when we were providing museums with Audubon Core exports of all of their stuff so that the museum could then include that Audubon Core of MorphoSource, of basically metadata from MorphoSource records. They could put that with their IPT, and then iDigBio and GBIF could pick that up. We have not made that. What happened was we shifted from the 1.0 version of the site to this 2.0 version of the site. And it was neat. I think a few, University of Florida used it. I feel like we didn't see total adoption of it. And so I feel like we've been wanting to sort of rethink how that might possibly work, whether it makes sense. I know you don't want duplicate publishers of data, but whether it might make sense for for MorphoSource to somehow publish those Audubon records if it didn't produce data duplication in GBIF or IBIC Bio. It's something we'd like to do. I just think we need to think a bit more to try and get it widely used.
Seems like it would increase discoverability a lot.
100%.
Yeah,
Yeah.
100%. One thing we do is provide our collection managers with that manifest of deposits. And then I'm not sure about the workflow on their end, but my assumption would be that they connect that with their collection or their specimen record, and hopefully that gets harvested by GBIF. So hopefully it does happen, but I'm not sure about that workflow.
Yeah, and I'll just, one thing I didn't talk about is we do everything at this point, a pretty flexible, fully featured API, so that if any institution wanted to, basically any institution that wanted to start putting their MorphoSource media in with their GBIF record, I think could very easily accomplish that using the API, or even like an endpoint researcher, if you're using GBIF to pull down occurrence IDs, you can use the MorphoSource API very simply to sort of look for specimens in MorphoSource with those occurrence IDs. Again, it's not the same thing as, but I do think there are also options out there already.
Mike, did you have a question?
Yeah, well, so first, thanks to all the presenters this morning. Some super exciting and interesting talks. A question for Julie. So you said that use the imaging event as a way of bringing together institution, instrument, and object, right? You also have multiple objects that were scanned in the same imaging event, so you can have a one-to-many, so one imaging event. There are lots of objects that are associated with that one.
Yeah, I think that's, yeah, I think that's a great question. And it's an answer that I have to answer, one on the level of the data model, and then the second on what can be done in MorphoSource today, which is we built the data model intentionally thinking about, you know, potentially even many-to-many relationships that you might have 20 physical objects scanned at once, and then you might produce, multiple media from that. I think what we're able to do in the site today is a little more limited, but you could actually in the site today have, if you had 20 images that were 20 physical objects that were imaged separately, and then a digital composite was made, we can do that in the site right now, where you have object image, and then the imaging events tie into like maybe like a digital composite or something. So that is possible with a little bit of special doing.
But the over project, there are a bunch of things that are crammed together in a can and all.
Yeah.
So at the same time, right?
Yeah, and I will say and this is getting, my opinion is that doing a burrito scan like that is necessary for efficiency. Yeah, where you take like a tube and you put like 12 fish inside it. I think any solution where you are trying to reposit that entire combined image without individual crops of each specimen, I feel like that's a recipe for confusion. I feel like it's a recipe for data being lost at some point along the line. Because if you have a tube image that has 12 specimens in it, and that's all you have, I feel like no matter how much you try and provide metadata alongside that image, you are inevitably going to produce a situation where someone confuses which specimen is which. That's why for those, we tend to be pretty insistent that users crop out each individual fish into its own, CT stack before putting it in the site, just to avoid that strong possibility of confusion.
Yeah, you could imagine though that in one day when a whole bunch of things were imaged at the same time, there was there was a loose wire in the machine, and you want to be able to pull all those out really easily and figure out which ones are imaged that way. But the reason I'm asking those for a different reason, a lot of different data and specimen types can be collected on an expedition in a collecting event, and being able to tie those together as a single collecting event is really important for a lot of reasons. And it just strikes me that your imaging event is the analog of that.
That's true. That's true.
A question for Julie also. We upload a lot of our CT scans for our papers to MorphoSource, like everyone. But something we worry about a lot is how apocalypse-proof MorphoSource is. So right now there's funding, and it's great, and it seems like a great thing to put in our data availability statements for our papers. But we're already running into a situation, not with MorphoSource, but with journals, where you can kind of supplementary material link from 10 years ago, and it's completely broken and goes nowhere. And so we're kind of worried / interested in what is the future prospectus for that? How's MorphoSource going to be surviving in the future?
No, yeah, it's a very reasonable question. For the long version of this, my colleague Doug Boyer actually at the ID Biodigital Data that was a couple weeks ago in Lawrence gave like a 15-minute talk about MorphoSource's sustainability plan. So I'm assuming those are recorded, and I definitely think that's worth going to look at. But the short answer is, between current funding, like we have commitments in hand easily out through 2029, if not longer. And we have never assumed that we were going to receive permanent funding from NSF or from Duke. I feel like both NSF and Duke were very, very clear with us from the beginning that, you know, that funding will be provided to help you get to a point of sustainability. But you know, we've never assumed that the only way MorphoSource is going to exist is through that. And one of our guiding principles is that if a digital resource is useful to a community, the community must be willing to help keep it going. That it's basically like, you know, that what is valuable to people will help keep going. So we plan over the next 5 years to grow a consortium of institutions and other larger organizations who would have an interest in helping financially sustain MorphoSource in exchange for a voice in governance, but also probably access to some power user features and, you know, things like that. But we are also thinking, you know, you don't want to plan to fail, but we've always been thinking of transfer of governance, transfer of data, and, you know, what could you do? And I think part of sort of the federated node model that I mentioned, where, you know, groups that were interested in spinning up a local version of MorphoSource might help spread the data load out and help achieve that. But thanks.
There's a kind of an optics thing with the with that international project. Well, I remember talking to some folks at the meetings in Australia this year, and they said what we really want is kind of a MorphoSource, but for Australia,
right?
For Australian museums, and it seemed like a strange thing for me that they'd have to reinvent a thing that already exists. But I do think there's that sense of ownership, you know, and national ownership over a particular thing. So a MorphoSource.au or whatever would presumably achieve that without having to go back to the drawing board and do that as well. Is that something you've bumped into in your travels?
Yeah, so we actually do have some Australian collaborators that we're talking to about Australian MorphoSource. I think in terms of collaboration, which I think we're super interested in. Yeah, I was mentioning we have a couple of different groups in the UK, one in the heritage science sphere over there to mention that, who have a proposal in right now that would see them spinning up, again, a federated instance of MorphoSource. It's not forking the resources, it's making sure that every node can see the data and the other nodes. But that would be, if that were funded, it would see MorphoSource as a United Kingdom national repository for heritage research imagery data. Okay. We also have a collaboration ongoing with the Natural History Museum in London, because they have conveyor belt scanners producing petabytes of data for them to spin up a local version for managing their things. And I think those collaborations are just a little more mature in the plans, but we also have conversations with Australia, because I think we have to recognize there is a bit of a cultural difference between how repository infrastructure is handled in the US from how it is seemingly starting to be handled in some Western European nations, and maybe in Australia, where there is more of an interest in the idea of like nominating national repositories or something like that.
Okay, up next, Kit Lewers. Thank you all so much. Yep, up next, Kit Lewers.
Make sure this is still. Okay, perfect. Hi, everybody. My name is Kit Lewers. I'm at the University of Colorado Boulder. I'm also at IDIG Bio, and I work with NASA on their Bioscape Biodiversity survey campaign. I'll talk about that a little bit. So my research really focuses on how information overload impacts institutions grounded in big data, specifically looking at how that creates disciplinary silos. and how certain architectures like microservices, event-driven architecture can help alleviate some of that burden of sharing data across a lot of different disciplines. I won't be talking about that too much. What I'm talking about today is basically the basics of information ecosystems and probably stuff that you all already know, but it's more of like gentle reminders of when you're thinking about things for your pitch and also about things with data in general. So yeah. So what are information ecosystems? So they are basically a dynamic network of interconnected data sources, tools, technologies, and stakeholders that facilitate the collection, management, sharing, and utilization of information. And these ecosystems are characterized by diverse data sources, which can include databases, cloud storage, sensors if you're working on remote sensing, and other data repositories to provide varieties of information, but still in a cohesive way. Interconnected tools, so that would be like your software applications, devices, and platforms that process, analyze, visualize, store data. And different stakeholders. So we have already a lot of really awesome fields represented in this room and way more represented in the broader field of biodiversity. So those could be things like researchers, they can be scientists, analysts, policymakers, and the public and citizen scientists that actually interact with the data are all people to think about too. Data flow and exchange is also a part of an information ecosystem. So that is the continuous movement and integration of data across different components that enable access. And as we know, software is changing, data formats change, and this really has to be thought about so we can proactively respond to how the state of the art changes. And then dynamic interactions. So I kind of already said this, but the evolving relationships and feedback loops between data sources, tools, stakeholders that drive innovation and inform decision-making. So these need to be designed interoperably to ensure that various systems and applications can work together to enhance collaboration and maximize the value of data, which then gets turned into information. Okay, so also, interoperability, so ability of different systems, organizations, and applications to work together seamlessly. The concept is crucial in data management as it ensures diverse data sets can be shared, integrated, and utilized effectively across platforms and disciplines. And the main way that I've seen this and that I specifically work with on my projects that I'm involved with are fair data principles. So those have already kind of been covered, so I'll just kind of go, but yeah, findable, accessible, interoperable, and like findable, we talked about this a little bit, but the use of unique identifiers and rich metadata to ensure that different data sources can be not only discovered, but also linked to each other in meaningful ways, because there's a lot of context that like people in biodiversity understand that when I like hop over to the remote sensing community, they're like, what is this and how do I find it? So that's really important, making sure that it's accessible through standardized access protocols and authentication methods. If you are using APIs, make sure that you can make those as relatively easy across disciplines, and even using industry standards if that is possible, so you can foster that collaboration. And then also making it reproducible, so comprehensive documentation, which has been talked about. Clear licensing, that's a big thing, especially if you're working with anybody in the EU. Licenses are extremely important to facilitating not only finding that data, but being allowed to use that data for future training in machine learning, presentations, and things like that. So those are things to keep in the forefront of your mind. And like I've already said, this really improves the long-term value of data. And I loved what Julie said, she's not in here, but we're thinking about how data can be used in 200 years, not just 2 years from now. So this is all infrastructure that we keep in the back of our mind. Like I said, nothing groundbreaking. But one thing that, at least in information science, we always say is you only notice infrastructure when it is not working. So make sure that you're thinking about it before it gets to that point where it's like, oh, why isn't this working now? So yeah, and also this is an AI generated image. I thought it was really cool. So I don't know what most of these things are. Satellites don't really look like that, but I really liked it. So wanted to bring attention to that. So, okay, it's cool. So another thing that is really important to think of, and this is something that I think of a lot in my research on microservices architecture, is not the fact that half of my word is missing, but that says intentional and scalable design. Something got messed up with the formatting, but intentional and scalable design. So standardization and interoperability. So it's once again, adopt those common protocols to ensure seamless data, and then the scalability and flexibility. So design infrastructure to scale efficiently with growing data and user demands, and try to predict how you cannot incorporate problems that are specific, but just problems in general. So for example, in microservices architecture, they are technically infinitely scalable. And you can ask me more about microservices. I'm really passionate about them. They're technically infinitely scalable because there are certain issues in biodiversity and remote sensing that we don't even know to think of yet. But we need to build an infrastructure that can incorporate those and keep incorporating them, but also allows for other users to continue to manage their data in a way that continues their research, but allows other people to come to the table when necessary. So having that scalability and flexibility, data quality and security. So quality control is something that everybody's already thinking about, and robust security measures, I think, are going to become more important as we go forward. Just to make sure that we don't lose the data. I'm not talking like, nobody come look at my data. I don't want you to share with me, but more like, how do we make sure that if there is a significant failure, what are we going to do to get that data back? Because there's just so much like rich stuff in museums that have already been digitized. And that is a very labor intensive and time intensive process. So it would be a real shame to lose that effort thus far. And then user-centric approaches. So we need to think about who is using it, not just the person who generated the data and the need that they solve for it, but how can other fields get a piece of the pie and use it? So talking to the communities, not just the researchers, but the policymakers, the citizen scientists, and see like what design concepts make the most sense for them engaging with it. And then of course, collaboration and documentation. I know I've said documentation so many times, but there's been so many times that I've been on a GitHub repo and there is documentation and I look at it and I'm like, what is going on? So making sure that your documentation is not only understandable to your community, but also to communities outside of your own. Okay. And sort of how this is being put in action is the NASA Bioscape project is what I'm working on specifically to figure out how we can incorporate a diverse community of scientists, citizen scientists, and an international audience. So currently what we're looking at, this is NASA's first biodiversity field campaign using multi-scale and airborne imagery. Please feel free to talk to me about this. I will talk about it forever. But we have 19 teams just on our project and we are already like, oh my gosh, like we don't know what this team is doing and we don't know what this team is doing. And this is just in our one funded area. And we kind of started facing the problem of how do we make sure that a global community of researchers know what we're doing and know how to access our data, and it doesn't just stay in our little data cube on our NASA server. And some of the data that we're working with and trying to get out there to different organizations are general remote sensing data, so like temperatures, humidity, things like that are pretty normal. We have a bunch of eDNA from air samples, water samples, and soil samples. We have AVRIS NG, which is next generation imagery. So that is hyperspectral, LiDAR. There's also thermal data, like just this giant, massive chunk of flight lines that go over and take really awesome imagery. And then we're also fitting all of this into satellite imagery and using field observations to kind of validate what we're seeing at the AVRIS level, and then using the AVRIS level to then validate what we're seeing at the satellite level. And it's just a lot of stuff. And we have a hard enough time making sense of it. And we really want to make sure that we are building infrastructures in a way that people can take pieces of it, but also see the whole. So that's kind of what I'm working on. And also, I just really want to say, like, think about how other people want to use and interact with your data. It's very important to think, like, what is the purpose that you're using it for? But there are probably things that nobody has thought of yet that, like, this vast this vast collection of data can be applied to. And machine learning is obviously a big one, but I'm sure there's going to be many things in the future that we can use it for too. So yeah. I'll read this off of my computer because apparently my formatting got really messed up. But conclusions and just a couple of things to like think about as you're going forward and talking in your groups and designing things for this potential expo is make sure you're thinking of networks of interconnected data sources, tools, and stakeholders. Try to see how you can enable seamless integration and collaboration across systems and disciplines. So those are social systems and technical systems. Facilitate data exchange and enhance research outcomes across various fields. So think outside of your field if you're able to, or bring people into your group that aren't in your field. And create user-friendly and effective ecosystems through standards if you want to like make a couple of crazy standards, or we have a wonderful consortium of standards from Tadwig if you want to pull those in and see how you can use those to make sure everything is standardized. Think of data quality in a collaborative culture and design and implement interoperable information ecosystems in your field, to enhance collaboration and research efficiency. Because that's the other thing that I forgot to mention is in a lot of fields, but even just like specifically in our project, we realized that a lot of people were doing the same analyses to go to the same point, and it would have been so much faster and so much more efficient if we would have kind of co-opted together and be like, I'm going to do this one time, we're all going to work on it together, and then we can really start making more inferences and conclusions. So that's the gist of it. Thank you so much for your time, and if you have questions, come find me or email me. Ed Stanley from the Florida Museum.
Yes. Oh, thank you.
On the back.
Okay, gotcha.
One second, Ed. You'd be good to go.
All right, thank you. Yes, so I'm Ed Stanley. I'm from the Digital Imaging Division at the Florida Museum of Natural History. And talk a little bit about maybe. Do I have a?
You should point it here.
Oh, I see. I'm sorry.
I might need to click on it. Let's
Oh.
try that.
Oh.
1 second.
No problem. That's okay. At some point, I'll be talking about CT imaging and how to share that data.
Some point soon, hopefully.
Working. That's fine.
No, it's
I could just say slide old time style.
it's not letting me change it either, so that wouldn't work.
Okay. It's my with my PowerPoint, no doubt.
Maybe try that. Okay, there's one more thing that I can do. I'm so sorry, Ed.
That's okay. Usually I fall into the trap of putting a lot of animations in my slides, but I didn't do that this time. So hopefully it'll we can just exit out and advance it if need be.
Okay, try that.
Am I pushing the wrong button? I might be pushing the wrong button. No, it's the forward button, right?
Well, it's not letting me advance either for some reason, and I don't.
Well, how about just go to the next slide? We can just go to the next slide, and then well, I guess there is a little bit of
Okay. Okay. I don't know. Let me just try unplugging it and
how about that? There you go. That old anti-crab.
Can you try clicking next?
Oh, I just got a buzz from my from the thing, but I don't know if that's is there a laser?
Sharing it on a different machine.
Sharing it on this machine. It's not letting me move it.
It's okay if you want to just keep it on the thing, we can just slip through it, not get too far behind.
Okay.
I'm happy to do that.
I'll try to fix it for you. I just don't understand how things can work perfectly and. Would you be super upset if it had your slides on the side?
No, that's good. It'll give people some chance to work out how much time I have left to talk. Pace themselves appropriately.
Okay, sorry about that.
And all right, no problem. All right, okay, so we can go to the next slide if you don't mind. So I think over the last 20 years, we've done a pretty good job at turning our physical specimens into digital representations of themselves and their collecting events, including things like sound recordings and digitizing the collection events themselves in terms of latitude and longitude and field notes and things. And of course, the sequence data that comes from tissues that were collected at the same time. But for a long time, it's been quite difficult to capture the real physical object, the three-dimensional physical object, right? And so 10, 15 years ago, I think you'd look at publications that used large amounts of museum data, and it would be, things, locality information for GIS, because that's the data that was immediately and available to massively ingest, or things like GenBank data, right? But we've recently, through non-destructive 3D imaging like CT scanning and light scanning and photogrammetry, been able to kind of create digital facsimiles of the actual physical objects themselves. And as a result of that, there's been these repositories like GBIF and IDIG-Bio and Macaulay Sign Library and GenBank that are specifically for these things. And that's MorphoSource seems to be the one that has kind of emerged out of all of the different things. Can we go to the next slide? Thank you. So, it's been mentioned multiple times, but the Open Vertebrate project was envisioned as a kind of a way of supercharging some of these collections. So everybody's scanning their own thing and putting them up on MorphoSource piecemeal, but we decided that if we could create a kind of a broadly scaffolded sampling of the vertebrate tree of life and jam that up on online for people to use from collections around the US, that would help get this kind of formalized pipeline through. And so this is one of the pipelines that we've built for getting our predominantly CT data up onto the website. And if you can go on to the next slide. So MorphoSource has a pretty good, and Julie mentioned this a lot. So it's a pretty good way of building collections from multiple different museums and museum collections and compiling them into a way that is easily digestible and accessible. On to the next slide. But the trouble is that the data themselves are quite challenging, right? They're not as, even though it's this is a frog, right? And even on the cross section, you can see it's a frog. The way of getting access to that data isn't necessarily as clear as some of these other text-based things, for example. So these are huge data sets as well. These are gigabytes of data. They require certain specialist softwares to process them, right, in order to turn them from a series of cross-sectional slices into a isolated circulatory system or a skeletal system and things. So it's not as accessible as we'd like. And so when we were building this thing, we thought, well, brilliant, we've solved the problem. If we just scan it all and put it up online, everybody has access to it, no problem, game over, we can go home. But if we look at the data that's actually, the data for the people who are using this data, and you can go on to the next slide, you can kind of see that actually, just from the data reporting from the MorphoSource OVERT project, you can see that's a strange offset pie chart, but essentially about 66% of the users are from a university and another, let's say, 10% or so are from the museum community. So we're not really, the vast majority of people who are using this data are the people who are going to the collections you know, and doing this stuff themselves, right? We're not breaking in, we're not opening it up into entirely new communities, although that still leaves about it's a little under 20%, on the next slide, please, that focus on people who wouldn't necessarily have access to this data, right? So that's I think good. It's not, you know, it's not the kind of the blowing the doors off the collections and everybody floods in and uses it equally. But you know, again, at the same time, you need to have an understanding of, you know, some pretty complex anatomical knowledge, if not, you know, the methodological knowledge for processing these things. So next slide, please. I'm part of this research coordination network called the Knockdown Network that's trying to increase accessibility and apply these well worked over FAIR principles to computer tomography data sets from across all kinds of scientific and industrial fields. But no, my background's in museum work, and so we work with all kinds of people from all kinds of backgrounds. But I can go back and kind of apply the findings from all of these different resources back to our museum collection. So if you can go on to the next slide. So we're breaking it out into and applying these FAIR principles to data that's going up on MorphoSource. So for example, findability, in order to make your data discoverable, the first thing, and a lot of this is probably fairly obvious, but the first thing you need to do is actually, you know, regularly update your collection. So if you're scanning things, make that data, get that data up onto the data repository like MorphoSource as soon as possible, right? And we have developed batch integration or batch upload methods that have helped this. A lot of the trouble a lot of the things that make data quite hard to find is the fact that you're uploading things before the data is, you know, released to the public. So if you're wanting to upload your in-work projects, you can upload that as a hidden or a private file. But then when the paper or the research is made available, you forget to, you know, switch it over to an open source. So having some kind of automatic embargo release or a reminder or you know, even batch modification of accessibility is something that I think would help move that along, or at least just being on top of that, you know. Something that MorphoSource does really well is connecting with collections databases, but that does require you to make those connections, make those links as you're updating these things. So making sure that the data sets you're uploading are linked to IDIC-Bio, for example, really makes an increased ability to find those data sets. Having consistent anatomical vocabularies is useful, right? So if you're if you have multiple data sets that are uploaded as skull, head, and cranium, and you're using one of those search terms, you're not going to be able to find those data. So thinking about having some consistent terminologies, and then, you know, one thing we were a little disappointed about with the post over project is how it was still very, you know, most of the people using it were scientists and things, and we thought, oh, well, it'd be great for high school students and you know, artists and things. But of course, if you don't know the scientific name of a thing, you know, that makes it hard to find on the thing. So having some kind of supplementary labeling, common names, I know it's all nonsense, of course, common names, but having those as a supplementary label would help increase the findability of your data sets, right? And of course, you know, we need to be better as a community at making our resources, advertising our resources, and that includes, you know, linking on websites and other things, but also talking to the public and communicating with educators and our, you know, our university staff. Also citing, so MorphoSource does a pretty good job of having both DOIs and ARCs. Use those things, put those in your papers. You know, you can pull down that helps link things like. Okay, if you don't mind going on the next thing. Okay, close. So accessibility, and again, findability and accessibility, you know, are two sides of the same coin, as is interoperability. But the idea of having an open access download policy really facilitates the accessibility of these things. So there's, again, 3 tiers on MorphoSource. There's restricted, open, and private. So obviously private means that it's closed off, you can't even look at it, but open means that you can click on it and download it immediately after providing a reason for using it, but it's an automatic thing. There's no human element in there. Restricted data suggests that it pings the owner of that data, and then they say yes or no to download it. And that's kind of the traditional way that museum collections have been controlled. But it does add a delay in that accessibility. And also, if you're not, and we're all guilty of this, I'm as guilty as anyone, not super good at checking those things. They can go a day or 2 or a week even, and by the time you finally get around to okaying the thing, they've moved on, they've forgot, you know, they've got it from somewhere else. So making sure that what you can have open access is open access, and what you can't, you're at least on top of it. Making sure that the formats that you upload things in are I guess, not super proprietary. That's another useful thing. Also, if you're not interested in, or if you want people who aren't scientists to use your data, uploading the kind of the ready-to-go data, like meshes and even videos, works well, right? That increases the accessibility of these sort of data. We can also, you know, provide better and more comprehensive reconstruction methods trainings. And for those people who don't have, and most of us don't have laptops that are capable of processing these huge data sets, there are now coming down the pike these online resources that allow you to remotely access high-power computers for an open access software. So MorphoCloud is I encourage you to look into MorphoCloud as a resource for that. Interoperability, you know, again, these are diminishing lists because, of course, interoperability has all these other things from findability and accessibility, but standard scanning vocabularies and anatomical vocabularies, again, making sure that your media formats can open access, but also including some kind of standardization across your facilities, right? So using objects of known density that scanned alongside your data allows you to implement new cross-cutting analyses like density and things. And then thinking about how your data is going to be integrated and ingested into these large-scale AI processing functions as well is another critical thing. The reusability aspect is another really interesting aspect of MorphoSource, because I think they've thought about reusability as much as any other of these, probably more than any of these other FAIR projects. So the ability to download data and re-upload it as a derivative and link those things is a very useful thing, and you can build these ideas of reuse, and you can kind of track the usage of that data through its journey, through the derivative identifiers like the DOIs, but also what I'd love to see is better attribution or accreditation of the generators, right? The people who are doing the work, so you can show the downstream impact that your scanning and data sharing has had, and I think that would encourage people to be more open with that data. One thing that we didn't really think about much initially, but we're glad we did, is retaining the raw data, because new AI methods are really allowing to increase not only the resolution, but the signal and the contrast that you can get from the processed data sets, the tomograms, from, you know, frankly, not particularly good radiograph data. So there are some advances in that if you get rid of the raw data and just retain the kind of the working data, you're going to lose out on. And of course, there's a cost that goes with that, but you know, there's a trade-off with the access you have to those original data sets. And then I'm sure Julie will talk about the IIIF thing tomorrow.
But increasing the ability to work with the data directly from the websites, I think will make a big difference in how that's impacting the non-scientist folks. And then finally, I think doing a bit of curation of your data sets to present them in particular ways with particular appeal to educators, for example. So you can take, collections of data and package them for, high school classes or animators or have and build these packages that can then so the onus is kind of on the museums and the collections, I think, a little bit to make that data accessible to specific groups of people. It's fine to just dump everything on the internet and say have at it, but it's much more efficient, and you're going to get the results that you want if you can package and advertise these things in particular ways. So that's kind of my 2 cents on a lot of these things. Sorry, I ran over a little bit, but there's my acknowledgements, and I'd be very happy to talk about this in the break. Thank you very much.
Yeah, no, I did. It's like the, did it run out of batteries, maybe? It did pause. Oh, no. Steve, come on, see if you can make it work. It's my weak thumbs. I think that's what it was.
Working like.
Stand.
Okay, maybe I'm pushing the button too fast.
Okay, so up next, and we will have a break after this for Q&A, if anybody has questions for Ed, Kit, or Steve. Thanks so much.
Okay, great. So I'm just going to talk about a little bit of background on standards. And mostly I'm going to be talking about some of the recent innovations that we had in Audiovisual Core. And I'll just wrap up with a few implementation issues. So first of all, I tend to assume that everybody knows what TADWIG is, but not everybody does. So it's kind of a weird acronym that stands for Biodiversity Information Standards. It's a community-based organization that develops standards based on consensus. Probably the most famous of our standards is Darwin Core, but we also have ABCD, which also is for documenting organism occurrences. And then I'm going to be talking about Audiovisual Core, which was formerly known as Audubon Core. And then we have a new standard, Latimer Core, which is for natural history collections and others. So that's just a little bit of the scale within TADWIG. Audiovisual Core, as I said, is the international standard for biodiversity-related multimedia items. And there's a core vocabulary, and then we have also developed 5 controlled vocabularies to be used as values for some of our terms. The vocabulary is a living thing. We have a maintenance group that develops new terms and new technology based on community demand, and that's a little bit of what I'll be talking about. So the kind of key features of Audiovisual Core, it distinguishes between sort of the abstract media items and particular specific digital representations, which we call service access points. And another thing that some people aren't aware of is that properties within Audiovisual Core, if it's possible to express an IRI for them, we differentiate between terms that expect to have IRI values and those that expect to have literal or string values. It's intended to be representation independent, so we don't specify particular serialization methods, and that's something that's kind of assumed to be developed by the community. So the original Audiovisual Core was very focused on still images, and it had this idea that people would be creating these Audiovisual Core records that they would pass around as kind of like independent descriptions of media resources. In practice, it's very commonly used as a Darwin Core extension as a part of occurrence records. And there are also other standards such as CamTrap DP that map their terms to Audiovisual Core. And it's also frequently used for database fields or as keys in like API responses. So beyond this sort of original focus on still images, over the past several years, we've worked on developing some additional terms that can be used to describe sound. And in particular, the terms at the bottom are used for describing regions of interest within sound recordings. So regions of interest are another concept that we have formalized as a standard for describing particular areas within a media item. So those could be defined by spatial coordinates, like in a still image, X and Y, but also by temporal and even frequency bounds in something like an audio recording. So what that allows us to do using regions of interest is to differentiate between multiple occurrences that may be represented within a single media item. So for example, the image on the left was intended to be an image of a plant fruit, but it turns out it also has a somewhat unusual insect mine also in the image. And so with the regions of interest, you can designate or differentiate between these two. Similarly, this is a recording that is listed as a call of a broad-winged hawk, but it also includes a red-eyed vireo that co-occurs at the same time, but it has different frequency bounds. So with the regions of interest, a standard, you can differentiate between these things. So one of the problems with this is that by adding regions of interest, we've complicated the model. It's not as flat as it was. So we have multiple service access points for a single media item, but now we also can have multiple regions of interest within the media item. And one thing you may notice here is that the regions of interest are for still images are described as relative bounds rather than absolute pixel bounds. And that's because you can have multiple service access points that all have different sizes. So it doesn't make sense to express them in specific pixel dimensions. Another new development is standardized vocabularies for subject parts and subject orientations. So these are useful for searching, but also if you wanted to extract traits or do machine learning and you wanted to designate a particular feature, it's possible to do that now with these controlled vocabularies. The controlled vocabularies have a small number of sort of broader concepts. So for example, a right view has the broader concept, lateral view. Also, these concepts have been divided into SCOS collections to designate which concepts would be for subject part are appropriate for a particular organism group, or which subject orientations are appropriate for particular parts. So this allows you to do some possible validation, also to develop tools that would guide a user to select the correct concepts for a particular organism group or appropriate orientations for a particular part. So combining together these controlled vocabularies with the whole region of interest concept allows you to have some very rich description. So for example, a lot of times live organism images will have multiple organisms, but with the regions of interest and controlled vocabularies, you can specify each of the individuals in that image. Or if you want to designate parts of a specimen image, you can do that. Or even parts of a composite image that are in a publication. These are all possible now. The other thing which we are just finishing up work on is a content description vocabulary. So this is something that our users said they really wanted. So now, in addition to designating like different parts of organisms, we can also say, this is a color bar, this is a scale bar, this is a label, or this is an organism. And then if it's a part of an organism, you can further describe it with these other vocabularies. The other thing that we have going on is we have a 3D task group, which is headed by Doug Boyer, which a lot of you know, and it overlaps a lot with the other 3D standards groups. And so they are just wrapping up work on terms to describe capture modality, 3D resource type, resolution, dimensions, and surface quality. And the maintenance group is working with them to try to drag this across the finish line by August. That's our goal. So hopefully we'll have that in place soon. So I just wanted to wrap up by talking about a few implementation issues. So the big one is that Audiovisual Core is not being used in a lot of places where it could be. So a year ago, I did kind of a scraping on GBIV to try to find out how often the simple multimedia extension was used versus the more richer vocabulary in the Audiovisual Core media extension. And you can see that there's a lot of organizations, large providers that aren't using, they're either using the simple multimedia extension or they're just not providing metadata at all. And the same thing is true about museum collections. There's a lot of large collections that are not using the Audiovisual Core extension. So we'd like to get people to use that because obviously you can provide much richer descriptions of your media content with these different features. The other thing is that I just, in looking at a few records, I can see that there's some confusion about how to use Audiovisual Core. So for instance, people are using the terms that are intended to have IRI values with strings and vice versa. Also, some of the similar terms like title, caption, and description are conflated. So it would be nice to have some tools, perhaps validation tools that would help people make sure that their records are clean. The other thing is, like I mentioned, that introducing the regions of interest has introduced complexity in the model. And so we need to work out sort of standardized, like how you represent this in JSON or JSON LD. Also, maybe get in the new GBIV model, how you would represent regions of interest. So there's some work to be done on that by the community. And also it'd be great to have some tooling developed that would make it easier for people to use these richer tools. And also to integrate it with other things like IIIF and machine learning. So that's just a quick run through of some of what we've been working on. And I'd be excited to talk more about this with people in the future. So thank you.
We have a question in the Q&A.
Yeah, this is for Steve. William Kwan says, is there communication between CADWIG audiovisual and 3D teams and IIIF audiovisual and 3D teams? Some institutions use IIIF to publish their collections.
We have had conversations with IIIF people, and we have developed sort of recipes for like mapping. It's actually quite easy to just take these like relative regions of interest format and like multiply it by 100 and have the image API URI for IIIF. So it's the interoperability part, at least for 2D images and like IIIF is really pretty trivial. But yeah, we have like developed this with IIIF in mind, although we haven't, I mean, like our maintenance group is open to anybody. So we'd love to have more people involved if they're interested in that kind of thing.
And is there information about when the maintenance groups on the TADWIG website, or where would you find that?
So the Audiovisual Core maintenance group, I think we in the GitHub issue tracker, I think the meetings are announced in there. I hope they are. I'm not the convener anymore. Ed Baker is now, but we used to announce those on the Audiovisual Core issue tracker. And if you're interested, we have sort of like a mailing list, and I could put you on that as well.
I don't know if anyone's nominating people for questions. I do have a question, but I did also want to say in terms of, there was a question for that IIIF Audubon Core for the 3D stuff, and I'll just comment that, yeah, I think the other person heading up the 3D working group is my colleague Doug Boyer, with whom I work at MorphoSource, and I'm one of the IIIF spec editors. I'm going to talk about it tomorrow. So I think there's definitely there's some conversation going on between what's going on with Audiovisual Core and whatever will happen with IIIF 3D. But I did have a question, which is, so you were talking about tools to make features easier. We love Audiovisual Core at MorphoSource. We use it wherever possible. And I'm actually really excited to hear about some of these controlled vocabularies that I was not familiar with. Do you all have or plan to have like a simple JSON API for returning controlled vocabulary values? My model of thinking there is for describing cultural heritage object material, we use like the Getty Arts and Architecture Thesaurus. So we can pull an API when a user searches ceramic and it'll show up, their controlled values for ceramics. And I know your orientation and part are much smaller, but I'm just thinking if you add something in the future so we don't have to hard code it.
Yeah, so there are actually there JSON LD representations, and there you can access those on the TADWIG website. So those are not automatically generated. There's also like CSV versions of them, and so the actual list of terms documents are generated directly from those CSV files, so they're guaranteed to be like up to date. Whereas the JSON ones get built manually, but like at least in theory, you could point a tool at that and harvest those. I mean, the other thing is that those controlled vocabularies are intended to grow. Like the values that we have are the ones that we got from participants at the time. And so, as I mentioned in the beginning, this is like a consensus-based community thing. And so if the community sees something that's missing, we would love to add it. And there's a process for that. So please talk to me about it if something's missing, because we'd love to add it.
Cool.
Also, I don't know if this is helpful, but on my GitHub, there's a script that will scrape any vocabularies from TADWIG. It's called tadwig_scraper.py. So feel free to use that too. If that's helpful.
Yeah. I think what we would love to have, because that's the thing. It's like they're pretty short controlled vocabularies, and we could implement them and then update. But yeah, something like that where we could point it out a CSV that we know is always on, or if you all had a query tool, so that we could have a user enter LATE and it shows up lateral with a description, but that way if one day a new one shows up, we don't have to like go and recode it, it's like whatever.
There's a page on the rs.tadwig.org repository, a page that basically says, here's how you can get this in machine readable form. And like I would say the CSVs, everything is generated from those CSVs. So it can't be adopted and updated without it. So if you use those, and those are stable, that the URLs are stable, so.
Oh, go ahead.
So question for you, Kit, and then I guess maybe Ed as well. I liked your perspective or your emphasis on the user-centric view of making the data accessible. And I'm wondering if we talk about the raw data or data products. This is something that Ed talked about in his talk. It's really, packaging in a way that K-12 educators or whomever can use it, because they won't be able to use the raw data, right?
Yeah, so honestly, both. So this is something I've been thinking a lot about with like heuristics of usability and like people interacting with the data. So specifically with hyperspectral data, most people cannot interact with the raw format just simply due to like computer power, not knowledge or anything. But when we do bias corrections and when we do harmonization and processing, we are putting our own biases on it that may not work well for other researchers. So we, I think they're called L1, L2, L3, and L1 through 6 products is what we have. And one is the raw data, and then we have multiple iterations of processing. So people can kind of pick and choose what level and what ability they have to interact with the data and how much expert intervention they want to use. Does that answer your question?
It does, but I think there's a lot of scope for working with those communities to think, well, this is what you were talking about, work with those communities to figure out, well, what would be useful. And that's, both an opportunity, but also slightly daunting, because there's lots of communities out there and lots of different things you could produce, right? And so it's kind of thinking through how to best approach that is what I'm.
But I think the Merlin app is a fantastic example of that, right? You've got this database, this resource, and you've packaged it in a way that is very accessible. to a wide range of people, and it has a huge impact. I mean, I would say that's probably the most successful spin-off from a digital data collection of any, because it's just, it's buttons you push, listen to the birds, and it uses the resources and tell you. That's a hugely impactful example of packaging this kind of data in a way that makes it accessible and useful to people.
I think in that example, that's because we know that community very well and what they want. And so we were able to do that, put ourselves, because a lot of us are part of that community, so we could put ourselves in those shoes. Likewise with eBird, the big change for eBird, which is a data in thing, not a data out, but still same idea. It was very unsuccessful for years until we took ourselves out of it and stopped thinking about, well, what do we want? Instead, we thought about what's that community want. And that's when it turned around. And that's challenging to do.
And I think like one big thing is making sure that you have appropriate mechanisms for feedback. So people can get in touch with you. Like it is a labor intensive thing to make to do human computer interaction and make sure that things are working. So I know like specifically for like me, I do a lot of interview studies and like do open calls and try to make myself available. So I'm not just like, this is what this log is doing. This is how people are clicking through it. So this is how they want to use it, but being like, how are you using this? Because this is what I'm seeing and what's missing for you.
We're ready for Will Weaver next.
All right, good morning, everyone. My name is Will Weaver. I'm a PhD candidate from the University of Michigan. Today, I will be talking about how we're expanding Leaf Machine 2, and we use Leaf Machine 2 to extract as much quantitative trait data and useful information as we can from herbarium specimens. So this is an overview of today's talk. We'll start with a little bit of motivation. We'll discuss the training data that we use for Leaf Machine. We'll go over the workflow and then talk about some of our expansion examples. So I've always been fascinated by the sheer diversity of leaf shape and leaf architecture in angiosperms. These are figures from a 1975 paper by Leo Hickey and Jack Wolf, where they grouped taxa by leaf architectural features to identify phylogenetically informative patterns and relationships. And James Doyle revisited this in 2007, adding molecular data into the mix, and ended up revising some of their conclusions. These analyses used fairly basic discrete character matrices, but in the age of AI, computer vision, machine learning, big data, we have the ability to look at a lot of these relationships at a much larger scale and with more informative morphological data. So one example of where I'm getting my morphological data from is the humble herbarium. Herbarium vouchers are widely used in biological research. We can get genetic data from them. They're useful for their historical record. We can use them to track invasive species, and you can even use them to inform conservation status. If you want to dig a little deeper into any of these topics, here's some papers I would start with. So the three on the left are things I've worked on. Papers on the right are really great papers from other researchers. But ultimately, all of these efforts can be broken down into three main tasks for using machine learning in biology. We want to automate labor-intensive tasks. We want to mobilize large data sets and ultimately generate a lot of useful data that we can do cool research with. And that brings us to Leaf Machine 2. While developing Leaf Machine 2, we've learned a lot about what it takes to have good training data. When creating our training data, we prioritized the inclusion of diversity for every modality that we could. We also chose to create a modular machine learning framework based on a delegation strategy. So breaking down a complex problem into solvable, smaller components. Our goal is to make generalizable algorithms, so our training data needed to reflect that. In that vein, this diagram outlines the machine learning modularity of Leaf Machine. Each box is a machine learning network of some kind. So green are object detection algorithms, cyan are large language models, gray is scene detection, Orange are key point detection and white is instance segmentation. The output from all of these models is eventually aggregated so that we can get a pretty holistic picture of all of the information that's contained within herbarium specimens. Over the years, we've had to manually generate a huge amount of training data to make all of this possible. We've annotated more than 500,000 plant components inside images, more than 200,000 archival components. We've manually annotated about 250,000 landmarks in different leaf pictures and manually segmented more than 30,000 leaf outlines. So that brings us to a happy more than 1 million annotations, which I think is pretty exciting. And I want to thank all of these folks for helping make it possible to annotate all of those specimens. This is the basic outline of Leaf Machine 2, but there are some parts that are not included in this diagram. The first thing to notice is that our plant component detector and archival component detector numbers 2 and 6 drive the entire process. They're responsible for creating bite-sized pieces that we can then analyze with component-specific analyses later on. So let's follow this specimen through Leaf Machine. We start by identifying 11 different plant components. That includes things like ideal leaves, partial leaves, buds, fruits, flowers, roots. The bounding boxes isolate each component so that we can process them individually. Once we have isolated ideal leaves, for example, we can send them to be processed by our instant segmentation algorithm and landmarking algorithms, which looks something like this. We currently use Detectron 2 and Point Rend to generate our segmentation masks, which isolate the leaf lamina, petiole, and holes in the leaves. And since our process is modular, we don't have to waste time processing leaves that aren't suitable for this kind of analysis. We also detect 11 different landmarks to form another set of measurements, which includes tracing the petiole, measuring the apex and base angles, and things like that. Stepping back, we can overlay all of this onto our final quality control image so we can see what's happening, and we get a nice, pretty image like that. And here I just wanted to highlight the segmentation ability of our algorithms. We trained our algorithms to ignore things like mounting tape, which greatly increases the quantity and quality of the segmentation max that we can generate. So even though our algorithms are pretty good, the whole point of this project is to scale up the size of the projects that we can use this kind of trait data for. So we've also been working on automated quality controls using things like visual question answering large language models. We've developed a few different methods of doing this, but essentially we can show the large language model one of our output images, like the image on the left, give it a set of instructions and a rubric, and it can output a score and an explanation of that. So in this case, it shows that there's a minor mask emission. We can see a tiny little bit at the top that it wasn't able to detect. So another example would be something like this. In this case, the bounding box cut off the very top of the leaf. It assigns a lower score and gives us an explanation. In this case, there's no mask at all, so we get a score of 0. In our last publication, we tested Leaf Machine across all angiosperm families and found acceptable performance for 245 out of 340 families. This figure also outlined the next step in our training data journey, where we've begun to fill in the gaps to increase support for the poor performing taxa. A few weeks ago, we incorporated the first tranche of this new training data, which included several thousand taxa that have large leaves. And we ended up reclassifying a few of the classes, and this resulted in a huge performance increase. So I'm pretty happy with our trajectory on that. And at this point, we can start looking at the archival components in the images. We identify 9 different things in the image. So that includes the label text, rulers, barcodes. And just like with plant components, we can isolate them and process them individually. We process the rulers using several different computer vision and machine learning algorithms. Our goal is to have some sort of way of cross-validating pixel distance into metric distance. You can see here, we were able to detect millimeter and centimeter marks and come up with a conversion factor. And we calculate this image specific. So every single image gets its own conversion factor. And if you've seen me talk about Leaf Machine before, then you know that I have a love-hate relationship with herbarium rulers. These are some of the rulers that we've encountered in our project, and I'm here to say that some are just better than others. If you're in charge of digitizing a collection, just use one of these two rulers, please. Or at least a photogrammetrically compatible ruler. If you want some data behind that plea, this is a comparison of human-generated conversion factors to autonomously generated factors, and you can see that these two are just objectively better and more consistent. Finally, we can talk about labels. With Leaf Machine 2, we can crop out all of the text and create a label collage. And then the goal is to do something with that. And there's a lot you can do with the text inside an herbarium specimen image. But basically want to extract the text and reformat it in a computer-friendly way, which is typically a JSON dictionary. And this portion of the project has kind of become its own thing. These 14 institutions have been collaborating with us to help turn this into a curatorial tool with the goal of digitizing a lot of the labels in these specimens. That's a huge bottleneck in herbarium data flows. If you're interested in the project, you can go to this URL link, which is a tiny URL/vouchervision, but it's cut off down there. We published a little essay about this already, but now we're scaling it up and doing some fancier things. Basically, the simplest approach to get a large language model to transcribe a label and reformat it into a JSON object is through prompt engineering, where we provide all of the context and instructions, including the OCR data, to the large language model. This works pretty well. It's easy to figure out what's working and what's not, but the problem is it can be pretty inconsistent. A second approach that we're taking is fine-tuning large language models just for this task. You basically have some instructions, show it unformatted, unstructured OCR data, teach it to create a JSON object. This method is really efficient. Once it's trained, you can just show it raw OCR text and you'll get the JSON object out the other side. A third approach that we're working on is probably my favorite. This is an AI agents approach, where we use multiple large language models that work together. We have a supervisor and a worker. The supervisor manages the project essentially. And for every label, it delegates to the worker feeds back, and you get a self-correcting loop, and you can even watch it correct errors that it's done. So this is just a really cool approach. And then finally, you can do all of this in one step. There are multimodal machine learning models these days. So things like GPT-4o, Gemini, they can accept text and images. If you give it the right prompt and the image, you can directly go from an image into a JSON object like you see on the right. The problem with this is it's very expensive to do this. We're working to make it cheaper, but that's a really expensive option. So you can try this on Hugging Face right now. You can upload whatever image you want. It usually only works for herbarium specimens right now, but you can upload anything and see what happens. Yeah, so that's all I have. Thanks.
And next up, we have Quentin Bateux, who is a postdoctoral associate here at the Yale Institute for Biospheric Studies.
Sorry, we had figured it out on her computer and now we have.
Oh, yes. No problem.
Okay, I did put a few animations, so.
I thought it was just my weak thumbs. Just a soft push.
Yeah.
Okay. Alright, so I'm gonna talk as well about biomes. And here I'm gonna talk about a very straightforward technique that could be helpful when you are doing a classification task to actually get more out of your data out of current models. Can we go to the next slide? Yeah. So I'm not gonna go into much detail about Hereld specimens. It's been talked right before. But yeah, kind of important resource to be used has been more and more digitized. So we have a lot of those in museums and still quite a lot online nowadays. Go to the next slide. So here I'm going to talk about kind of a trust mechanism for automated labeling. And so the task I'm talking about is a classification task. For example, if you're interested into the reproductive state of a sample, the question you're asking usually is, are there flowers, fruits, buds, nothing, all of those. Next slide. Sorry, I did animate. So for these tasks, there have been people looking at doing classification models from deep learning to that automatically. And they had decent accuracies on the 80–85%, depending on the tasks they were looking at. Go to the next slide. What happened is, although this paper was pretty nice and the model was out there, the actual usage of this model was pretty low because from a bottom standpoint, when they look at the way they are doing their workflow right now, they're working with people, students, volunteers, where people have shown that the accuracy of humans for this type of task is about 97%. So botanists look at that, they look at this gap, 85, 97, and the conclusion is like, it's not ready yet. Just wait and just continue doing research as we are doing right now. Next slide, please. So I want to show here one very simple mechanism that can be used to push a little bit and squeeze a little bit more out of this specimen by using a trade-off. This trade-off is possible because most deep learning classification models give you not just one class, like I think there's fruits, but they will give you a probability out of the softmax layer that will be more or less accurate, but overall it will be accurate enough for our purpose. It's going to say, okay, I think there are fruits at 60%, or I think there are nothing at 5%. Next slide. So the normal way, the mainstream way to use these models is you take the biggest probability in there, and you say, this is the class I want. This is what the model is telling. This is where you kind of flatten everything into the model thing that are fruits. And so you annotate every single pot of data, and you end up with your 85% accuracy in top one. That's a very straightforward way to use this model if you want to annotate everything. Next slide. But what you can also do is recognize that for some tasks, for example, classifying large amount of data that is already there, you don't necessarily care about annotating everything. And if you realize that, that means you can have a rejection mechanism where you actually set a threshold saying below this confidence of the network, I'm going to assume the network is not sure enough, and I'm going to put this label into the unsure category, just ignore it for now. And I'm going to just keep everything that is above this threshold. And if the network is not too biased, and if the probability is reliable enough, that means that you're going to see an increase in the performance of classification of everything that is left. Go to the next slide. So just a very simple example here, just to illustrate that. Say you have these 4 specimens. You run a classification model through that, like saying flowers or no flowers, just this binary model. And it's going to give you a label and an associated probability. And here it's wrong for one of them. And turn out that it's the one for which the actual probability was lowest. So what we do, if we go to the next slide, is we actually can say we can try different confidence thresholds to try and see if this intuition of the higher the confidence requirements, the higher the probability. And if we do that we take one model, we take some data that is for which we know the actual truth, so we can actually test the performance of the model on that. And we can actually move this confidence threshold from the lowest, which is everything above 50% is true, up to like nearly 1 for which we trust and nothing. What we see is that what you see on this graph over there, in red, it's the amount of data we reject. So it goes up, that's kind of normal. The more the stricter we are, the more we reject. But interesting, what we see is in blue, we have the performance, the accuracy for the model for everything that's left. And here we do see this monotonous increase. The more we reject, the more accurate we get. And we can do that for any model that's out there on the internet. And then we can get this graph, and we can look into this graph and compare that to what we need. What is our requirement for that? Get to the next slide. For example, let's say you want human accuracy. You can look at the graph and have an idea, okay, for this particular model being trained on this task, if I want human accuracy, I can expect to reject a little above half of my data. What that should be at about human accuracy. And to get that, it gives you this threshold value, which I showed you the threshold value about. It's like 97% accurate certainty for the model. So you can actually find your own trade-off for your own task in there. Which is the next task. So we did some testing on that, and the main part in there is a study replication. So we start from this study that was working on New England specimens, which were the model that we trained was. So we are as close as possible in terms of domain. Same specimen, same species, same area. So they looked at like 15,000 specimens, and they were very interested into the relationship between invasive and endemic species, and looking at what was the fruiting season differences. The nice thing with this species, with this study, it was pretty straightforward to actually find the exact data they were working from and actually get the images. We can go to the next slide. So we found the images and we ran these images through our automated model to give us like this fruit, no fruit, and follow the methodology of the paper. And so if we just take the normal way of using the model, like taking the main class, whatever the probability, this is what we get. So here we have a bunch of different season estimation for fruiting for a bunch of different species. And in blue, that's the ground truth from the study. We now assume that it's the most accurate data we can get. And in red, this is what the season spread you can get from the automatically annotated data. So it's not amazing. Uncertainty is really high. It gets quite shifted. And most importantly, if you look at what they were interested in, like if you look at native invasive species, look at the fruiting season shift, you find 9 days compared to the, yeah, we cannot see it right here, 26 days that the study found. So quite a weak result. And it seems here botanists are right, not in the goals right out of the bat, because it doesn't really look quite good results. Let's go to the next slide. But then if we apply the method and we say we want human accuracy, even though we get into kind of a worst case scenario here, because we are necessarily working with less data than they were, because we start from their set of data, this 15,000. So everything that we reject, we're gonna get less data than them. But still, we apply this, the threshold where we expect to have 97% accuracy. We lose a bunch of data, about 70% here. For 3 of the species, we do so many that we can actually even get the flowering season, the fruiting season. But for everything else, now we are way better aligned with the findings they had. Like most of the, so we are just way closer. And when we look at this final information we're looking for, this shift between flowering, fruiting season, we get 25 days off. 25-day shift, so 1 day off from the actual study itself. We are into something that seems way closer to actual reality there. Let's go to the next slide. I'm gonna click there. So we tried it as well for not just binary classifiers, so not just yes/no models, but for something with more multi-class that we found at random on the internet, basically trying to find as little input from us. So we found something trained on iNaturalist 2018 that was trying to do some classification and recognize what species was in a given image. So at the end, it gives you a big list of 8,000 probabilities, thinking, I think this is this species in this image. We're going to go to the next slide. And when we do the exact same analysis from this random model we found on GitHub, we can see the exact same trend. So the model has a base accuracy of like 42%. And we see that we can actually bring it up to about arbitrary values at a cost of rejecting some of the data. But we can actually still apply the same methodology there and get potentially for a task, if we don't care as much about coverage, but more about accuracy, we can actually find a middle ground that's closer to our needs. And next slide, last one. So yeah, just in conclusion there, it is possible with the models we have right now, even if they seem not as good as we hope them to be for application, we can actually still annotate at least a part of the data we have available, which is quite big nowadays, and still get quite a lot of this data annotated at a level of accuracy that makes it usable and research ready to make like studies that are not in the 10,000 specimens, but hundreds of thousands or millions of specimens right now with the current models. Yeah, thank you.
And our last talk of the day is Rob Guralnick, who's the Curator of Biodiversity Informatics at the Florida Museum of Natural History.
All right, guys. Well, great to be here. As we get the slides set up, maybe I should try telling a joke. Also, I recognize I'm standing between you and lunch, so hopefully we'll get this on the road. And I purposefully have kept this presentation relatively pithy on the grounds that we might be a little bit off our schedule. So, okay, so the joke, right? So, why can only dogs be radiologists? So let me try this. Let me make sure I get this right. So why can't dogs? Why are only dogs radiologists? Wait. Let me try this right. So, why can't dogs be radiologists? It's because only cats can. Come on, come on. That's good. You know, I tried up here. All right. So when I was thinking about what I would do for this presentation, I started a kit, and was like, "Kit, what the heck should I do for this thing? You know, what would be useful for the audience?" And she said, well, talk about like linking and harmonizing traits across data ecosystems, right? And so that's a pretty interesting challenge, and that's going to be the focus of what I talk about today. And what I want to do to start is talk about this from the perspective of a data sharing ecosystem. So I love thinking about data sharing ecosystems. Kit mentioned this as well, and my take on it might be a little bit different than hers, but kind of aligned in a bunch of ways. But what I want to do here is really focus on this ecosystem, which revolves around people. People are at the heart of this ecosystem, right? And people are at the heart of technical parts of the ecosystem as well as the people part, right? So like, to kind of walk you through it, you know, we can think about primary data producers. These primary data producers have content that they have. They want to make sure those content are discoverable and usable, interoperable, harmonized, right? Which is super important. And typically, these primary data producers are working to form these data sharing networks, right? And so, for example, you can think about the thematic collections networks that were developed through the ADPC and IDBio efforts. And those data sharing networks really collaborate to make the data sharing easier, to kind of share knowledge, to share practices, to work on an interoperability challenge, right? And those data sharing networks are producing data that have to go into technical infrastructures. And really to make that work, what I think is fundamental here is to think about like kind of three key parts of this technical infrastructure in the middle of this diagram, right? Which is data and metadata standardization, the importance of having these access tools, whether we're talking about APIs or compute software that wraps up those APIs so people can access this content. And we need these integrated data stores, which are complicated to build, as many of us have talked about the challenges and complexities of building these data stores. And you talk about cloud storage, or whether you have a document store, or a relational model. There's all kinds of complexity here. All of this is really hard. And then there's also hard parts with just managing taxonomy and doing data QA and QC. All this is like a big challenge, right? And then equally challenging is making sure that user communities know how to use the data that you're producing in these integrated data stores, right? So you have your user communities are diverse. You have communities of educators, you have communities of scientists and decision makers and managers. They all have different needs, they all have different understandings of how to access this content and use it. And they're all coming at it from different angles of what they're trying to produce at their end in terms of knowledge, right? I think it's really, really cool and interesting what's come up a lot in the discussion today already, which I think is super fundamental to the world we live in today versus the world that I lived in 10 years ago, is this right part of the diagram, right? These secondary data annotators and the loop between the secondary data annotators and like AI and ML approaches and computer aided approaches, and humans and computers like working together and making new content, right? And the new content is really, really interesting. It's kind of where I want to lean on this talk, because that really creates challenges with linking data. It's come up a lot among all of the presentations about how hard it is to kind of get this pipeline working between the secondary annotators, the computer annotations, and where those go into the integrated data store. If you're familiar with iNaturalist, most of you are, there's a whole pipeline of annotations that are happening all the time to improve identification. So identifiers come in, they help with making user-grade data through identification processes. All those annotations are stored in this data store, or the annotations can go off somewhere else, but have to link back to the data, the specimen record data that you're collecting, right? So this whole pipeline of secondary data annotations is really, really exciting and challenging and interesting. That's where I want to talk about linking and harmonizing trait data that come from specimens and then are basically annotations that need to be linked up and maintained, right? So, 1 second here. Many of us have lived in an ecosystem where this works, right? This is a virtuous ecosystem. It requires thinking about credit models and equitability. It requires thinking about inclusivity and fair data, right? But one of the things I was thinking about a lot is how difficult it can be to link. So for many years, I've had conversation after conversation where someone would ask, like, well, how do I link a specimen to a GenBank, a specimen data record to a GenBank data record? How good is that ecosystem? How good is that linkage? It stinks. It's pretty bad. So we can't propagate a lot of the time, and we can't link up the output from an assay that comes from a specimen to a repository that's storing the outcome of that assay, like a gene or a genome, right? So that linkage is really hard to do. And the problem, I think, is literally about this, and Kit mentioned it as well, right? We basically silo our data infrastructures pretty firmly. We don't mean to, but it's really hard to avoid siloing. So many of you are kind of nodding your head that silos are like kind of what we do. We don't do it on purpose. It's not like we're intentionally trying to keep data in silos. It's just they kind of form from the fact that our ecosystems can be little worlds unto themselves, right? But what I want to make a case for is that we can think about our systems and like, what if we think about our systems as sort of this idea of a global knowledge graph, right? And a global knowledge graph is really powerful because, I'm sorry, it's cut off a little bit on the bottom here, but it basically says, you know, what do we think about our systems as global knowledge graphs? And what's exciting about a knowledge graph to me is like, imagine you have a single specimen, right? And you want to like look at that specimen and ask, what are all the data that have been generated from the physical specimen? Where's the specimen record? Where's the CT scan? Where are the genes? Where are the traits that were measured off of that CT scan? And pull on that thread. And when you pull on that one specimen thread, you pull all the content, all the threads that are connected to that specimen. To me, that's a powerful idea, right? And so I think instead of a single disconnected box, we can replace it with a really simple view. We have a central resource, this middle blue circle, but it's connected to other circles that are themselves connected to other circles to build that graph, right? And how do we make it work though? It's really challenging to build this. If silos and virtuous ecosystems are what we build, this is even harder, right? But at heart, we need identifiers, globally unique, persistent, shared identifiers. And we need semantics. And the semantic layer is the hardest layer of all to build. So what I want to do is show you an example of how this can work. I'll make it quick. So this is called Futurist. And it's a funded project that's focused on basically taking measurement data from specimens and linking it to those specimens and providing access to those measurement data, which come from a whole bunch of different disciplines. And so what was really exciting to me about Futurist was the project was funded to basically work between paleontologists who measure bones, archaeologists who measure, that's what all paleontologists do. So if you're a paleontologist in the room, you're not just measuring bones, but like a lot of it's measuring stuff. Archaeologists who measure stuff, and neontologists who measure stuff. And a lot of neontologists measure external measurements of the specimen, but there's all these different kind of measurements of traits that happen, and they all go in different spots. And they all have different worldviews on how those measurements happen. There's London Trench measurements, there's measurements that paleo people do. It's all very different. And so what Futurist is all about is basically building the ecosystem around linked data, right? So how does this work? Well, before I get there, why do we do it besides the disciplinary integration and the ecosystem building to make a more global knowledge graph? This is a great quote. I won't read it, but I will say that a lot of trait data in the world is at the species level. So there's all this species trait data. I love it. It's great. But the problem is, if you want to get to the questions that really matter on global change, it's the individuals and the populations that matter. Aggregating up to the species limits our ability to do the core science we need to do to understand change. Whether you're thinking global change from the recent perspective, or you're thinking about global change from a time extended perspective back into the macroevolutionary patterns and processes, right? So getting to this individual level data is incredibly valuable. So how does Futurist work? Well, basically, we help contributors publish millions of existing trait measurements. These are mostly external measurements for neontology, but for paleo and archaeo, we have a lot of data coming in from people measuring bones. So contributors have 2D measurements, they have 3D measurements, they have image data from the literature. Those all go into a pipeline that basically starts with people providing spreadsheets. We annotate those spreadsheets with ontology terms that allow us to make data interoperable, and that goes into the Futurist data store. We help contributors standardize and harmonize how traits are reported and measured. And what's cool about Futurists is that the ontology gives us a lot of power, which I'll explain in a couple minutes. Okay, so a pipeline for ingest. We have a pipeline for people to discover and use our data in a fair way. So we produce tools to help end users discover and best use these trait data. And we put a lot of those tools to basically encode best practices. We want people to use best practices in terms of discovering these data and knowing how to use this data appropriately and effectively. And so we have visualizations and lessons and maps and resources that make it so that people can get these data and use them the best possible way, right? Okay. We also, because we're focused on linked open data, we also can really integrate data and reduce silos. So we have the ability to link the trait measurements directly to different large-scale resources like IDBio or GBIF or Open Context or the Neotoma. Open Context is for archaeodata, and Neotoma is for paleo and archaeodata. Probably these all have their different kind of emphases. But the linkages mean that these measurement data are always linked back to these repositories, right? Which is super important and handy. And I'm really excited about this link between Futurists and being able to use community science tools. I'll just go with the fact that if I go close enough, it kind of changes. But there's a real link here between basically leveraging the community science angles around coordinating trait measurements and linking into Futurists, which I think is really exciting. And then finally, just a quick note on this ontology framework, then I'm done. I'm really, it was a huge amount of work to do this, but building these sort of application ontologies around what we call the FOVT, which is a vertebrate trait ontology for measurements and data, and the BCO, which is basically observations and measurement oriented. These can help basically take data from different kinds of measurement practices, whether we're talking 3D traits with landmark data, we're talking ordinal traits or linear traits. This can all be integrated into this pipeline using, again, a whole set of tools like linked open data approaches and GitHub that allow us to basically get to this like really sort of linked open data framework data repository built. What makes the Futurists ontology really powerful for me are two things. One is really easy to add new traits. So if you have a relational data model, it can be really hard to like rejigger your models. Ontology models allow you to kind of Lego brick them up. So you can kind of add new traits and make it really easy to do that. What I think is also really important is we can actually use a branch of machine learning called machine reasoning to kind of reason over the data and produce new data that are inferences that give us power. So if I wanted to go and ask a repository, like, for example, let's see, like show me all the traits you have on forelimbs of all kinds, whether it's ordinal or 3D morphometric or linear, give me all those data. Normal systems can have a hard time giving you those generalizations. On an ontology framework, we can ask that question, we get back all those data instantly, right? Because the ontologies can generalize information about different kinds of measurements, knows that an ulna and a carpal and a metacarpal and a radius and femur all come from the same forelimb or hind limb, and can kind of do those generalizations for us, which gives us a lot of power for searching and its discoverability, which I think is really cool. So my pitch here is linked open data, thinking hard about building out repositories that don't silo your data, and kind of building out ways so we can link this up in an ecosystem that's larger than any one single ecosystem is, sort of at the level of the global graph, the big semantic web. Check out Futurists. It's a great project. It's been fun to work on. We're actively working on more, and it really reflects to me not just a one example of a repository, but more of a way of doing business, a way of thinking about the world that will make it better and easier for all of us down the road. So thank you.
Well, yes, so we do have a few minutes for questions. I did want to say to folks who are virtually in attendance, we will be back here tomorrow at 9 a.m. At Eastern time to focus on highlighting innovative technologies for data use. So I hope everybody can join us there then. But if we do have any questions for our last few speakers, please feel free to put it in the chat or raise your hand if you're here.
Okay, I'm sorry. Hi. So Rob, hello then. One thing that museum collections tend to do quite poorly, I think, is record collection information about the environment that the specimen, the immediate environment the specimen was collected in, right? And I think now with field photogrammetry and three-dimensional capturing of data, we can actually generate some really cool data-rich media to help pull data out of that. And so it feels like there's a bit of a, you know, a tabula rasa, like blank slate. We could, if you were going to sort of build a system where we're starting to collect that data in a meaningful way, do you think that do you think it's a kind of a several small or single large, you know, is there a centralized way to do that fits all of that, all of the media, all the kind of collection events that we're building? Or do you think, is it the sort of thing where we just say, you should do this and we'll worry about piping it all together afterwards? Does that make sense?
Yeah, that's a $1 million question, right?
Yeah.
Like, the more we jury rig these solutions post hoc, the more we end up with a whole set of like unexpected problems. Like that's caused a lot of grief about trying to like think about retrospective versus prospective, like what you can do with retrospective, like linking of data versus like what we can do now, right? I prefer to get it right up front.
Sure.
On designing this, right? And I mean, linking those data and images directly to like vocabulary, so it come from like ENVO, for example, the environment ontology, which might from peer visual events, and thinking about how all that will be part of like a larger ecosystem related to like collection events, which will presumably be like a linkage that a collecting event will be linked to the environment which the collection came from.
Yeah.
I mean, I think I push back and say like, although I think what Austin's doing on like field photogrammetry is like really, I'll use a Californianism, bitchin', or rad. I think there's a lot of habitat data that's been captured on labels. I don't know if people on the Librarian side look at this a lot, but like there's tons of habitat content on many, if not most herbarium labels that is super relevant for capturing sort of contextual knowledge about what happened during the collecting event. Those are like field notes, right?
Sure.
So all this linked up is like really challenging and hard, but I prefer to think about what that looks like, at least at the framing of it up front. And like I think with Steve's talk, I noticed like you build it strong enough to be bulletproof against most of the things that you do, and then you start refining and digging in, right, and building more. And I think like it's so cool to watch like regions become part of like the Audubon core. I think we can just start with like a really core framing of what we absolutely have to have. And then we can kind of dig in and kind of grow out the like what would be awesome to do is this next thing, right?
Is the danger if you build, if you build it, pre-hoc, that you're missing stuff? You know what I mean? Like you say, this is the way you do it. And then like years later, you're like, oh, but there's some stuff on the periphery that actually turns out to be really important.
I mean, I would turn that back at like everybody, because I don't have an easy answer for this. Like every infrastructure, every standards development process kind of has its own, like people are just part of that process. And so it gets messy around the edges of what people need and want.
Yeah, that wasn't a question. That was a question to the ether, I think.
Yeah, I mean, I think that all development of infrastructure and networks like that has to do both. And it has to have a cycle. It has to have like an iterative development cycle where I think you need to put in good, serious thought, ad hoc, pre-hoc, to try and put it together, but with the recognition that you're gonna mess up, you're gonna miss things, you're gonna go a year in and then you're gonna slap yourself on the forehead and be like, how did I not see this? We desperately needed this. I think you have to build that expectation that you're gonna get a ways, and then you're gonna have to go back, and then you're gonna have to, and it's a never-ending process in a way until you're done.
But having that kind of forward-thinking view means that you're not doing it, you're doing it accidentally rather than just being, ignoring. I
think intentionality.
Yeah.
My question was, with the machine reasoning layer that you're using, do you think that it would be able to build some recommender systems that go across the disciplines and networks. So people who are maybe over here in that network can get recommendations from like over here that they're not usually connected with.
I think that's such a cool idea. Yeah, I love that. I love that idea. And I just don't know enough about the intricacies of recommender systems to know how that works. But I think ontologies are really valuable for helping. I mean, part of that's like there's sort of this push and pull for me. Like there's formal ontologies, which can be very complex, and they're axiomatic. Like you actually have axioms built into them that do logical calculus that help you avoid misinference, like transitivity properties and other kinds of ways that you really need those things to work to make sure that people can produce the right kinds of inferences from those data properly. And there's like folksonomities, right, which are really powerful and help to kind of connect data and content or tagging and the looser parts of that. So I just don't have the expertise to know what this bigger ecosystem is going to look like. But there's a lot of super smart people here who probably know a lot more than I do.
Question for Will. Maybe you said this, but what do you see as the next big hurdles and challenges for the direction that that project is going? What's the next big thing that really needs to be?
Duplication of effort, I think, is a big challenge. It's like I can process 100,000 images or 1 million images. If I change my algorithm, then what? If someone else has some other component, how do you integrate all of that? So I think that's a big step. It's also very taxa specific still. When I say angiosperms, there's plenty that we can't do anything with. Like if it doesn't have broadleaves or even for Poaceae, sometimes we can handle that, sometimes we can't. There's also a taxonomic limitation too.
Did you still have a question, Steve?
In the interest of time, I'll ask Rob in person.
Are there any other questions at the moment?
That's a quick question. Well, it might be a quick question. It's definitely a naive question. I heard Mally Hudson give a talk on biocultural labels last week. This is coming out of the CARE position repair, and Indigenous knowledge has the potential to be represented both in the nocturne activities and also in this kind of futures project. Have you thought at all about biocultural labels for stuff? Is that part of the data model?
I feel like people get quite squirrely with this. You have to tread so carefully with it that it's kind of a can of worms where almost wait until we feel comfortable solving some of the low-hanging fruit, which of course is worse in a way to just say, well, that's too difficult to manage, because of course there's solutions, but it requires coordination and networking with folks that we don't have those connections with just yet. So that, I mean, it's something that we're not deliberately ignoring. We're just trying to get the kind of the frameworks in place, the kind of the low-hanging fruit that isn't as challenging taken care of first, so then we can apply the lessons learned to those situations. I think certainly from the nocturne side of things.
It's a really critical question. And what I've had the honor and pleasure of like working with archaeologists who think a lot more about this intersectional spot. It's provenience and sort of site context, so what it means from people perspectives is so critical for the foundations of the discipline. I feel like that's been a set of lessons learned. Like I think from a neontologist perspective, we have really rich biocultural data, but we don't know how to really deal with those questions well. Yeah, and care principles, which I love and I think are super important. They're just hard to like figure out the edges of implementation and kind of building out that larger sort of space for the work to happen. I think it's something missing because I look around this room and we just don't have like maybe out of our skill set, to be perfectly honest, to know exactly how to move here, and it requires broader community participation to seal out these questions. And there's like questions a mile deep here. I think Ed's right on the money. And part of it is just like as you go down further into what you're thinking about, it gets further and further and more complex. And I think probably at Indigenous knowledge at a cultural level, the complexity is actually much higher than like a collecting event and a sample.
Any other questions?
Stop. Recording, 23 for